Right. So then we'll start the session, right? Good afternoon. So it's up to speed sessions day 3. Okay. So now at this point, even I think there's no need to introduce the program that is happening. And at this point, I think the people have also been filtered. People who were there in early days are no longer joining. That means they have been filtered, which is natural. Okay. So I always want to have a balance because I'm not going to keep waiting until everyone is catching up. Got it? Because this is a English medium biology class. Therefore, we have to work towards a result. We have to work with a proper plan. Got it? So everything I do, I have to maintain a balance between making you understand all the concepts, but also I have to go according to my timetable. Got it? I am not going to keep on waiting until everyone has caught up, until everyone out there is going to join my class. I'm not going to wait like that. On the other hand, I'm not going to go fast as possible so that I can claim, okay, we are doing the third lesson now, we are doing the halfway in the third lesson now. I'm not going to do that. Okay. So if you have been joining the regular theory class, you can understand so in Saturday class, I just finished photosynthesis, but there's a small part remaining to cover. Okay. So in 2 more weeks, almost 3 weeks, we'll be able to finish the second lesson. Okay. Then we can move on to the third lesson with a brand new set of tutes and a brand new set of question tutes and everything. Okay. So you can understand that is the proper speed to go. Okay. I have been doing day papers with you. I have been doing past paper discussions with you. I have done practical discussions with you, practical demonstrations with you. After all those things that I have done, I am at this point in the syllabus. Got it? I am not too fast or not too slow. That's what you have to do. Otherwise, I can't go to my plan of covering the syllabus in time. Okay. So if you are doubtful if I'm going to cover the syllabus in time, you can analyze, see whether if I'm going in a proper speed, you can understand. I always say you can go to other classes, you can go to all whatever free sessions happening in other classes and see, right? I have told you this is still the time for you to go and explore, go and try out different teaching methods out there, different ways of learning biology is out there. But at the end, you have to keep in mind this has to go with a proper balance. Got it? The syllabus has to be covered, but at a proper speed so that it's not an express train, and as well as it's not a slow, very slow process where at the end the syllabus coverage is going to be not properly done. Okay. Tute, I think so. You can send a message to the hotline. Okay. So last Thursday, got it. The tute should be there. Okay. So you can drop a message. I have sent a number in the messages I send for all the tute issues. You can send a message there, but if you can't find that, you can send a message to the hotline as well. They will solve all the tute related issues as well. Okay, website related, payment related, tute related, all that. For that, there's a dedicated person for me and my classes. He's almost always online, so you can contact him. Okay, don't worry, it will arrive. Okay, 2 things could be there. It is the fault of the Sri Lankan postal service. The other thing is could be somehow could be an issue due to, delivery issues could be there depending on certain issues with your address also. So those will be solved. So you can understand, you are just starting this program. Got it? So you are new to this process as well as, so once you have started receiving 1 or 2 tute sets, the whole thing will become a very smooth process. As with my 26 batch, 27 batch, they are all accustomed to this, the whole website as well as receiving tutes and everything. Okay, so I'll start with our plan. So we have today's up to speed sessions day 3. Okay, listen, today's up to speed sessions day 3. And I'm planning to have, so today, including today, I'm planning to have 3 more, 27, 3rd and 10th. Okay. So 27th, 3rd and 10th up to speed sessions are going to happen. Listen carefully now. Okay. So up to speed is taking a bit longer than I want to. It's not going to the speed, but that is good because this program was designed to so that I can bring you all into our normal theory class time, the theory class pace. Okay, so including today, so today is 20th. Okay, so up to speed is going to happen on 27th as well as 3rd August as well as 10th. Okay, up to speed sessions. Then our normal theory classes are happening now. Then the special announcement is there's going to be, once the up to speed sessions are over, at the same time in the theory classes the second lesson is going to be over. Chemical and cellular basis is going to be over. Got it? So once the chemical and cellular basis is over, there's going to be a term test. All right, more than a term test, it's actually end of the lesson, but I'll give it a name. It's going to be a term test. Got it? So that is going to happen for the two batches. Thursday batch on 13th, Thursday, 13th Thursday at the normal theory class time. Okay, so it's going to be a short paper, maybe 1.5 hours. Got it? Then for the Saturday batch, it's going to happen on 15th. Got it? So I'll try to deliver the paper to you. Online people, listen up. I'll try to deliver the paper to you, but you have to make sure that you open the paper only at the exam time when I tell you to open it. Tell you to open break the envelope and take the paper out. Got it? So I have been doing that sort of exam for my 27 people. So now it is the time to start for you. Okay, you have to join the Zoom meeting and then you have to open the exam paper envelope. Got it? So online I can't exactly monitor you, so you have to have some self discipline. Got it? So the term test is going to happen. I am going to give actually two papers. I mean, two papers means two different papers because I'm going to put out the PDF also. No? So on 13th Thursday, the paper is going to be out. So the Saturday people have a chance to get the paper earlier. So there will be two papers. Two papers means two different papers. Got it? And the papers at this point, I haven't exactly decided the structure. There is going to be one SCQ at least, one essay. Okay. Then about 15 MCQ is what I am planning. So let's see, I can put up to maybe 20 MCQs. Okay. So you don't need to worry. Okay. You don't need to worry. Got it? Because up to speed, there's a lot of time until the exam, and also from the up to speed sessions, anything you have missed will be completely covered. Got it? Now you can understand, right? You can understand in the theory class, we have recordings, about 16 recordings to cover the content I'm covering in about 6 up to speed sessions. You can understand that, right? In about 6 up to speed sessions, I'm covering the content that I did in maybe 16 classes over 4 months. Okay, so it is going to be much easier. It's easier means you are on a, let's say, not a shortcut, but it's a fast express pathway. So take the maximum advantage of that and be properly confident before you get the exam. And for the online people, there's going to be top scorers. Okay, I'm going to choose the highest scorers for the Thursday batch, right? Top 10 for the Thursday batch and top 10 for the Saturday batch. Physical people, there's going to be top scorers. Online people, there's going to be top scorers. Got it? So that's the plan. Okay. So don't worry about this. Okay. I'm not going to make the exam very tough, but it's an exam. Okay. An exam is an exam. So you have to study for it. Okay. At the same time, just every day study a little bit, keep in touch with whatever that is happening in the up to speed sessions or in our regular theory class. Don't let it get collected. Okay, even the second lesson is a big lesson. Got it? So therefore you cannot, I'm trying to build your mindset. Okay, I'm not trying to sort of get to elaborate with it. I'm not going to show you some magic tricks here and there, okay, just to keep you attracted. But all I want to do is for you to make sure that you understand. At the same time, make sure you are prepared to face the paper. But that doesn't have to be a boring process. That's why I always try to keep your interest in the subject, because if you have a sort of photographic memory of the things that you are learning with the visualization, with some nice color diagrams and everything, it's much easier for you to do this for a long time. It's a marathon. I always keep on saying this is a marathon. You have to run for a long time, not fast for a short time. You have to keep running at a steady pace for a long time. You have to study for a long time to take all that knowledge into the exam. Got it? Because you will understand, like the 26 people are right now, my 26 batch, they are doing their final revision. There's only about 20 days remaining to the exam. So you can't absorb all the resource book, there's about 400 pages into the memory at these final two stages. But on all you can do in the final 2 to 3 weeks is just refresh it. Just refresh all the hidden hidden old memories of the syllabus. You can just take it out. Okay, that's what I'm trying to do. So that there is, from the beginning, I'm trying to put all these things into your long-term memory, so that when you are close to the exam, you can just refresh them and go to the exam without any stress. Okay, clear? So keep these things in mind, and so you can take out the tubes. Where did we stop? We talked about structures of protein. We talked about structures of protein. Did we finish all 4 levels of protein structure? Did you finish all 4 levels of protein structure? Yes, let me know. There are 10 units in the syllabus. Okay. Where did we stop exactly? Let me know. Let me know. Keep the chat active, with not with spam messages. Okay, right? That's, you can ask anything. Got it? Where did we exactly stop? Today we have to continue from denaturation onwards, right? Yeah. Okay. Okay. So at the same time, I want you to have the resource book on your side, and I need you to have the tute, because we have to complete the tute as we go along. Okay. We have a lot to cover today. Got it? I want to cover maximum content today, so that you are prepared before the exam. Okay. Okay, take out the resource. We'll come to proteins. The topic we'll start with is denaturation of proteins. Okay. As I always keep telling you, keep a nice separate color for definitions. Okay. So there's a definition here. Denaturation of protein is the loss of specific chemical 3D shape due to alteration of weak chemical bonds and interactions. Highlight. Denaturation. This is a definition, okay? You have to write exactly what is there in the resource. Right? You have to write exactly what is there in the resource. Methana, what are the keywords? If you take a protein from the tertiary structure onwards, now listen, from the tertiary structure onwards, you start to have a precise, unique, functional. These are all very important words that we use to describe the working level of a protein. A protein that is at a working level, a functional working protein. Got it? So, a working protein is going to work only if it is going to maintain its unique, precise, compact, functional 3D shape. Due to some reason, if this within a specific 3D shape is altered or changed, then that protein will no longer work. Imagine in a factory, there are machines. Your proteins are like machines. Got it? If you damage some of the machines, can the factory function properly? No. The cell will not be able to properly function if the proteins are damaged. You will see proteins perform so many enzymatic function, defensive, transport, storage, receptor, hormonal, so many functions are performed by proteins. Got it? If they change, the function will be almost always lost. Okay, so highlight this. You, as people who are studying biology, it is very important to know what can change this specific structure of protein or what can cause denaturation. Write down, you need to know 4 main categories of agents of denaturation. You can write down in this space. Okay, high temperature and high energy radiation. You can see that has happened here, no? When you are cooking an egg, right? This fried egg, no? Right? You can see the egg white, which is usually transparent, containing the egg white protein albumin, okay? Albumin. When you heat it in your pan, it's going to change its specific shape like this. So, original structure is like this. Can you see it is slightly changing, so that at the end there is going to be this white color part in the fried egg that you eat. So, this egg white, you know, in a raw egg, it is water soluble. You can dissolve it in water. But can you dissolve this cooked egg white in water? No. That is because something has happened to the structure of the protein that has changed how it behaves. Okay, its nature, its properties were affected. High temperature and high energy radiation could have an effect. Then, what are the chemical agents? So, these are, you can say, sort of energy sources, no? Energies. Then, chemicals are there. Strong acids. Strong acids can damage. If you burn your hand in some acid, that means the proteins in your skin, in your hand, muscles, connective tissue got damaged or changed by the strong acidity. Okay, strong acids. Similarly, you can have, due to strong bases, alkaline. Strong acids, alkaline, and high concentration of salts. High concentration of salts. Salts can have an effect on certain bonds of the protein structure. You know, there are ionic bonds and there are disulfide bonds. Okay, those kind of mainly hydrogen bonds. Those things can change when you have these chemical species. Okay, those things can change when you have this sort of powerful chemical species in the solution with the protein. Third one, write down heavy metals. Heavy metals. Okay, you will understand heavy metals can act as a catalyst. A heavy metal, so you know, no, these sort of nickel, cadmium, those kind of metals you learned in O levels that they are acting as certain catalyst. No, what do you mean by a catalyst? That they sort of promote certain reactions to happen. So, when you have some heavy metals in the mixture, that means some damaging reactions can happen. I mean, this damaging reaction will be stimulated by the heavy metal, so that the protein will be affected. Okay. Then finally, things that can dissolve away the protein. What it completely dissolve away the protein, such as what? These are organic molecules. So, organic solvents can dissolve the protein. Organic solvents and detergents. Finally, what is alkaline means? Basic. Alkaline, basic. Acidity, basicity. Alkaline nature is basicity. Okay, quickly write this down. That's all about denaturation. Nothing much to worry about. Okay. People who just joined, we didn't do anything. We are just talking about denaturation. Okay. Up to speed sessions with up to speed karan with that now. Okay. So watch the recordings. Okay. If you have missed anything because of connection issues or something, okay, please watch the recording. Got it? Okay. Right. We'll move on to functions of proteins. Very important. In your resource book, you have a this a chart. That chart is it's a nice source of uh, MCQ points. Okay, it's a nice source of MCQ points. So that's why I decided to give you all these diagram based, so that you can remember. You can remember based on the diagram. These things are the I mean functions and the examples that you need to remember. Got it? Alright. So I'm going to go with the order that you have in the resource book. So in the resource book chart, we are in page number. People listen. Page number 18. Okay. So we'll start with the catalytic protein. Yeah. So it's right. So enzymatic proteins, or you can say methionine. You can put a dash here. Okay. Enzymatic proteins or catalytic proteins. So if you guys joined when we were doing enzymes, you might have an idea what is done by an enzyme. An enzyme is a biological catalyst. That means it can promote a certain reaction to happen, usually faster, increases the rate of reaction, catalyzes. Okay. So catalytic proteins, within they accelerate chemical reactions. So you can highlight they accelerate chemical reactions. Okay. Then I want you to write down the examples that they have given in the resource. You can write down here. There are some space in it. Right. They have given pepsine. Then you can say amylase. Pepsine and amylase. If you have space, no need to exactly write this. Pepsine you can write in front. You can write in front of pepsine, digest protein. Amylase you can write down, digest starch. Okay. Just small, just to keep it in your memory. Okay. You can say pepsine digests proteins. Okay. Does anybody know where you can find pepsine? Where in your body do you have pepsine? Anybody? Pepsine koiti? Yeah, okay. Very fast. Good. Stomach. Amylase? Where do you have amylase? Okay. You watch my small YouTube shorts? Yes. I think those are not actually uploaded by me. There's a set of marketing people. Okay. Yeah. Subscribe. Better subscribe. Okay. Okay. Yes. Yeah, you are somewhat correct. Amylase is two forms, right? Amylase has two forms. There's a form that you find in your saliva that is called salivary amylase, and there's another form that is secreted by pancreas, pancreatic amylase. So the mouth amylase acts in the oral cavity. Then the pancreatic amylase usually acts in the small intestine. Okay. Yeah, very correct. All right. Right. So that is it. So catalytic proteins such as enzymes catalyze biochemical reactions. So in your resource, the second function is a structural protein. So we'll come here. Structural proteins. Okay. So in your resource, it's given as there are two examples. That is collagen is given. So for collagen, they have given the function. Can you see? Methionine, can you write down? Provide strength and support. Under collagen, provide strength and support. Provide strength and support. That is the function of collagen. Can anybody tell me what is the level of protein structure of collagen? What is the level of protein structure of yeah? Why do we say it's quaternary or fourth level? You can see here that there's a blue one, there's a red one, there's a green one. There are three polypeptide chains forming this sort of a cable. Collagen is a very strong fiber. Okay, so you take three protein sort of strands and then you make the collagen fiber. Then the other example that you have in your resource is keratin. So keratin is given here. Found in where? Within, can you highlight hair, horns, feathers, skin appendages? Okay. So you can write here Keratin will have function you can write down. Prevent desiccation. Prevent desiccation. I know the space might be a bit small. Okay. Prevent desiccation means usually it could be mainly water loss. Okay. So this keratin, because it acts as a sort of a hydrophobic layer, right? It can prevent water loss mainly in your skin. Okay. Right. So that is structural protein. Understood? We can move on to the next in your resource is storage. Okay. Now you have to work with me fast. Okay. Storage proteins, ovalbumin is given. So ovalbumin, what is the function of ovalbumin? You can write here storage protein in egg. Right? Storage protein in egg, ovalbumin, storage protein in egg. Then the other protein that is given is casein. Casein. So it is given here. Okay. Casein. So highlight that. Casein can you highlight here? And you can draw an arrow to it. You can draw an arrow to it like this and say storage protein in milk. Storage protein in milk. Okay. So try to sort of keep the tudes neat and the resource book neat. I know for the boys it's a bit kind of used to working in a just to get things done, no? But for biology it's going to keep it more interesting, and also you will feel like studying if your notes and the tudes and the resource book is a bit neat. Okay, so try to not beautiful but keep it let's say organized and neat. Got it? That's all. Right? I don't have best handwriting and I'm not a very neat person, but I try to keep it clear. Okay, so boys should try to improve that. Girls, of course, I think your work is sometimes too neat. Okay, to you sort of focus on beautifying it. Okay, so try to keep the notes nicely, easily. I mean, no, keep the notes clear. That's all I want. Okay, but keep it neat. So that I mean, why are you keeping it neat? Your class teacher is not going to come and check you. I'm not going to come and check your attitude, right? It's for yourself. Do it for yourself, because A level is a exam that is you have to do with yourself. You have to go with. I mean, you have to go to the exam by yourself. Therefore, I'm just a guide. Got it? It's not unlike O levels. Very unlike O levels. Okay. Storage afterwards, there's transport. So you can jump here. Okay, transport proteins. Actually, this sort of a transport is a different thing compared to the transport that is given in your resource. So this is a transport protein that we are going to very soon encounter when we are studying our plasma membrane. Okay, so you can call this transport protein as a channel protein as well. You can write here, this is a channel protein that is found in your plasma membrane, cell membrane, that can allow certain molecules to go through the membrane. But as those are not the examples given in the resource, but it's given in words here. Maybe hemoglobin. Okay, what is the function of hemoglobin? You can write here. You can write here what is the function? Transport oxygen and so did you know that hemoglobin transports carbon dioxide also? We always say hemoglobin is oxygen transport. So why are we saying transport oxygen and CO2? Why do we say that? Any idea? Any idea why we say that? That oxygen transports, hemoglobin transports oxygen as well as carbon dioxide. If you take the hemoglobin molecule, it is composed of how many protein chains. It is composed of how many protein chains? Yes, Ken. It is composed of how many protein chains? Yeah, 4, ne? The sort of protein, globular proteins, there's 4. Okay. They all have names. Okay. This is, all of them also have a iron atom, Fe atom, right? There's a Fe2 plus inside each of these. Fe2 plus, we call it as the heme group. Okay. So these all these protein pieces have names as well. This one alpha 1 and this is alpha 2. This is called as alpha, sorry, I'm sorry. This is called as beta 1 and this is called beta 2. Okay. So alpha 1, alpha 2, beta 1, beta 2, there are 4 subunits. Okay. So you already know oxygen connects to where? Oxygen connects to this heme group, no, to the Fe2 plus atom. Then where does the carbon dioxide bind? In the hemoglobin, there is a place for the carbon dioxide to bind as well. So imagine hemoglobin like a lorry, right? This is an example I like to use. Got it? Listen, okay? If you think of hemoglobin as a lorry, right? Okay? If hemoglobin is a lorry like this is the driver's cabin, okay? And this is the back of the lorry, no? So the back of the lorry is designed to carry oxygen. Oxygen පටවන්න පුළුවන් usually අපි කියමු මෙච්චර දාන්න පුළුවන් කියලා oxygen, right? But in this lorry there is space for carbon dioxide to go as well, but not in the place where you transport oxygen. Not in the place where you transport oxygen, but imagine it is with the driver. With the driver you can take some carbon dioxide. Okay. So there is no problem of the space. I mean, the carbon dioxide is not going to take up any of the space available for oxygen because it is going to travel at a different place, bonded to a different part. Actually, it is going to be bonded to some of the proteins, not to the Fe2 plus ion. Okay. So that carbon dioxide is also transported by hemoglobin. Okay. It's like a lorry that is transporting both, right? So keep that in mind. In future, it's going to be important for you. So transport oxygen and CO2 by hemoglobin. What is the other transport protein? Is it serum albumin? Yeah. Write down here as another example, serum albumin. Does anybody know what is serum? Does anybody know what is serum? Serum albumin, write down. The function, transport fatty acids. Transport fatty acids. Yeah. If you take our blood, listen now again. If you take our blood, you know, if you take some of your blood into a mehema tube, imagine you went to the, some of your blood had to be taken to do maybe a full blood count or some sort of blood test. Okay. If you keep this blood for some time, you will see the cells in your blood is going to settle down. Your blood has some cells, no? What do you have? You have red blood cells. You have white blood cells. Then you have platelets. All those are going to settle down because it's solid, no? The cells are solid. Got it? Then there we have a liquid part in your blood, right? You have a liquid part in your blood that is containing mainly what? What do you have in this liquid part? Plasma කියලා. Right? Plasma contains usually what? Mainly water. Yes. Then amino acids, glucose, yeah, vitamins, waste, urea, wageva, right? All those things. And in addition, there are things that you need for blood clotting. No need to think of this, okay? No need to think of this too much, okay? There are things called clotting factors that you need for blood clotting, okay? Nothing is out of syllabus. If I'm saying it to you so that you understand it better, that is all because it is in the syllabus, okay? So if you take plasma, if you remove these clotting factors, you have something called as serum. Okay, so serum, in this liquid part of blood that we call as serum, you can have water soluble protein that we call as, you can have water soluble protein that is called as albumin. That albumin is known as serum albumin. Okay, that serum albumin is very helpful to transport things that are water insoluble. Okay, very important to transport things that are water insoluble, such as what? Again, what are the things that are water insoluble? Fatty acids. Yeah, fatty acids are water insoluble. So you need the help of someone water soluble to transport it. Okay, that's why serum albumin transport fatty acids, because the serum albumin is water soluble. Okay, okay, right. Then signaling molecules or hormones. Okay, so you can see hormonal proteins. The examples are actually given. You know the what are the examples given? Okay, insulin is given. Okay, so you can see there's a lot of glucose in the blood here, and when you have insulin released, can anyone tell me? Insulin is released by the pancreas. Can you highlight this? Insulin is a hormone released by the pancreas. What is the effect of insulin? Very commonly known. Okay, up here I can write a small equation here. Okay, we can write a small equation here. Okay, where do you have space? Normal blood sugar. Okay, so I can write, so as the effect of insulin is already given, I'm going to write the equation for glucagon. Okay, I'm going to write low blood sugar. Okay, low blood sugar. Then there's another hormone that we call as glucagon. Either glycogen way, glucagon. It's a hormone. Okay, glucagon. Glucagon is going to do what? It is also going to bring blood sugar into normal. Imagine you have been fasting, right? So, got it, in your Ramazan period, people fast, right? And some people for their diet. So, to lose weight, people fast, right? So, in those kind of situations, you can have low blood sugar in your blood. I mean, the sugar or the glucose content could become low. So then, in such situation, you are going to release the sort of the opposite. අපි අර කියනවා antagonist of insulin. ඒ කියන්නේ insulin කරන එක අනිත් පැත්ත. He is going to do the opposite of what is done by insulin. That is called glucagon. Glucagon is going to convert low blood sugar into normal blood sugar levels. Okay, so මෙතන දාමු glucagon secreted. Glucagon secreted. Okay, write down high blood sugar, insulin secreted, normal blood sugar. Low blood sugar, glucagon secreted, normal blood sugar also. Okay, clear about hormonal proteins. Okay, then we'll move on to this contractile or motor proteins. These are able to generate force. These are able to generate forces in your body needed to, okay, sort of do your movements. Okay, so how does the body know blood sugar is high or not? There are some sensors. Okay, glucose sensors are inbuilt into the pancreas itself, because the pancreas is going to secrete glucagon and insulin. No, pancreas is going to detect. There are special transporters built into the pancreas insulin and glucagon secreting cells, and the glucose is going to enter into those cells, and the cells are going to themselves decide, okay, this glucose level is too high, then I'm going to secrete glucose. Then they are also going to detect, okay, blood glucose level is too low, then I'm going to secrete uh, glucagon. Okay, it is controlled by the pancreas itself. Okay, pancreas itself. Got it? Right. Contractile motor proteins function as they say. They generate the force necessary to move. It could be a single joint. It could be multiple joints needed to run or walk or climb or jump or whatever. How is it? There are two types of contractile and motor proteins in your body. That is actin. You can see when this orange colors are actin. Then the bigger one is myosin. This purple color. Got it? For now, you don't need to know how they actually work. You can see here, you can label this. This is not just any muscle. You can label this is skeletal muscle. This is skeletal muscle tissue. If you zoom into a skeletal muscle tissue, you will be able to see this sort of action, this sort of view of my actin and myosin arranged. You can write here contractile motor proteins function. Okay, contraction of muscle fibers. Okay, it's important that you study exactly what is in the resource. Okay, contraction of muscle fibers. Contraction of muscle fibers. Okay, right. So, any questions? We'll do a question. All of you, anything else remaining? Hold on. Defensive proteins are remaining. Okay, defensive proteins are remaining. So, we'll come here. Defense. So, I told you everything, almost every function in your body. I mean, your proteins are the workforce in your cells. They defend you, they transport things, they build things up, they catalyze reactions, they contract muscles. At the end, all the work in a cell has been done by a protein. Okay, your lipids could be there as structural to insulate or as energy. Your carbohydrates could be there as storage or energy, right? So, storage or structural. I mean, storage means later will be used to produce energy. So, their functions are limited. Can you understand? But if you take a protein, they are the workforce. They do all the work in a cell. That's why when protein making as well as protein breakdown, protein damage, all those are very important concepts. Got it? So, they say biology doesn't have a lot of mathematics or a lot of thinking compared to, let's say, something like physics. That is because the fundamental, the basis of certain physics laws is actually simple. You can apply certain equations based on original. Okay, so there are some discoveries. There's a Bernoulli's law, there's a Pascal's law. There are laws. No? So once that law has been established, there's an equation. You can apply to the equation, and there are some other assumptions. You ignore the air velocity, right? Then you can get an answer. Okay, you will understand how complicated bio is when in molecular biology we have to study how proteins are made. Okay, biology we have to rely on memorizing things because whatever we are learning is very complicated. We cannot go ahead without remembering what we did. Got it? You need to have in your mind the basic concepts. Otherwise, you can't understand biology. But in physics, if you know the basic physics sort of the law, then the equation, you can do a question. Okay, that is because it's based on a simple thought process. Okay, but in bio, it's a very complicated process. Therefore, that's why we have to memorize it. That's why we have to memorize it. If you don't memorize it, you can't understand the next step. Got it? Okay. So if you don't understand, next upcoming we are going to talk about DNA and RNA. If you don't understand how DNA and RNA make proteins, later in molecular biology, you won't be able to understand how we actually make the protein, how you modify them. Okay. That's why bio, we have to rely on memorization because if you don't remember the early concept, you can't understand the next step. Okay, that's why. So it's, I mean, that's why we say, okay, bio is all memorization. Yes, it is memorization. Why do we have to memorize? Because it's complicated. Okay, otherwise we won't be able to understand. Right? Right. Okay, come to defensive proteins. Okay, Metin, example, antibodies inactivate and help destroy viruses and bacteria. Okay, there's another name for antibodies I want you to write down, that is called immunoglobulins. Immunoglobulins. Immunoglobulins, write down. Then we'll write down what is given in the resource. Neutralize foreign bodies. Neutralize foreign bodies. Okay, listen. Your antibodies will be secreted. These are antibodies. These are actually pieces of protein. These are pieces of protein. Got it? These are pieces of protein. Then you can see here, there's a small structure. This is a virus. Viruses are very small. Okay. So once the body detects that you have a virus, could be the COVID virus. Okay. The body is going to start producing these pieces of protein that we call as antibodies or immunoglobulins. They are going to surround this virus and it will start to could be breaking down, could be inactivation. Okay, so the virus will no longer work properly. Okay, so if you take a bacteria is much bigger than a virus. Okay, so this whole thing is a bacteria, part of a bacteria. Okay, so bacteria is much bigger. So even then, antibodies will surround the bacteria and cause damage to the bacteria. The bacteria will not be able to properly divide and cause damage to your body. Okay, so that's it. Then there's a final one, receptor proteins, that is not given in the set of functions of proteins here, but we'll quickly talk about it when we are doing the plasma membrane. There are receptors protein in the plasma membrane that is going to sort of get involved in signaling processes. Uh, Yeah, there's a antigen and antibody relationship is there. Okay, we'll talk about that in when we're doing immunity. For now, remember antibodies are what? They are proteins that can help to neutralize. Could be foreign particles, could be viruses, could be bacteria. Okay, right. That's it. Those are the functions of protein. Set of protein functions. I hope you have uh, gotten everything written down, completed. Okay, right. We'll do a question and we'll move on to nucleic acid stuck gala. Okay, we'll do a question. I think we have some past paper questions neither. Mm, I'm make a ටක් ගාලා කරන්න බලන්න. Quickly do this. This, where is it? Ah, yeah. Proteins. Quickly do this question. Proteins. Quickly do this one. Quickly do this one. Everyone send me answers. Yeah, what's the answer? Right, okay. I think I gave too much time. Right. Okay. Proteins form secondary structure due to disulfide bonds. Okay, we'll write some small reviews. Can anybody tell me why do they form secondary structure due to? Can. Why do they form secondary structure? You have to mention properly. It is due to within the molecule, right in front. Intramolecular, intramolecular hydrogen bonds. Okay, not because of disulfide bonds. You can cut it with a red color pen or something, different color pen, due to intramolecular hydrogen bonds. Okay, intramolecular hydrogen bonds. Number two, made up of 26 different amino acids. No, you can cut it. Always when you are cutting and correcting. So, cutting and correcting is a type of writing a review as well. Okay, but do it with a different color pen. Okay, so how many amino acids do you need to make proteins? 20, no? Only 20. Are composed of carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. You don't need phosphorus a lot, neither. Only up to sulfur. Okay, only you need up to sulfur, not phosphorus. Would not be denatured by detergents. No, it is one of the main agents of denaturation. So, false. Contribute to transport of materials. I want to just write down some examples here. What are the examples that you need to know? Serum albumin and hemoglobin. Serum albumin and hemoglobin. Okay. Okay. Any questions? Okay. Right. Let's move on. Anything else that we need to do? Okay. Shall we move on to our nucleic acids? Because nucleic acids is going to take some time to discuss. Okay. So, we will get there. Only a small part remaining in this tute. I want you to, and I want to move on to the second tute as soon as possible. Okay. Let's come to nucleic acids. Anyway, everyone, all good? Neither? Right, not sleeping? Yeah, I'll give you a break after doing DNA. Okay. Okay. Come to the resource book. Have the tute on the side. Okay. Page number 18. Nucleic acids. Let's go. Nucleic acids. Let's see this. Nucleic acids are polymers existing as polynucleotides made up of monomers called nucleotides. Okay, so that's a, I mean, kind of a definition. So they are polynucleotides. I mean, so a chain of monomers. What is the building block? It is called as nucleotides. Okay, you need to remember they are macromolecules and biological polymers. So keep that in mind. They are polymers. Okay, highlight those. Then highlight the elemental composition: carbon, hydrogen, oxygen, nitrogen, and phosphorus. Again, he has given as macromolecules and biomonomers. You need to remember. Okay, out of the four groups of biological molecules, polysaccharides, proteins, and nucleic acids are macromolecules and biopolymers. Lipids are not considered as macromolecules or biopolymers. They are just some kind of a large molecule, but not big enough to be a macromolecule. Understood? Don't overthink it. Just big, not lipids are not macromolecules or biopolymers. There are two types of nucleic acids. DNA. What is the long name of DNA? Deoxyribonucleic acid. RNA is ribonucleic acid. Okay. So as usual, before we talk about the big polymer, we have to talk about the building unit. Okay. We have to talk about the building unit. What is the building unit? it is the nucleotide. You need to be able to draw a nucleotide. So I have given a molecular diagram of a nucleotide. You don't need to be able to draw this. You should be able to draw what? What should you be able to do? You should be able to draw the symbol of a nucleic acid. The symbol of a nucleic acid you should be able to draw. Okay, listen. It has only three parts. Very simple. There's a sugar that is called as a pentose sugar. There is a pentose sugar. There's a nitrogenous base and a phosphate group. Okay, those are the three parts. So I'll start drawing the nucleic acid. I'll start drawing the nucleic acid with the pentose group, okay, with the pentose sugar. So it's a 5 carbon group, right? It's a 5 carbon group. It's like this. The carbon atoms are arranged, listen very carefully. The carbon atoms are like this. This is an oxygen atom, okay? This is a pentose sugar. This is an oxygen atom at the top. You can see there also. It's an oxygen atom. The carbon atoms are here. The carbon number 1 is here, 2 is here, 3 is here, 4 is here. So where is 5? Where is 5? Yes. The 5 is going to be sort of extended. It is going to have this sort of an arm, and the fifth carbon is here. Got it? This is what we call as our, this is our pentose sugar. Okay. This is our pentose sugar. Then two other groups are connected to this pentose sugar. You have a nitrogenous base. Okay. Connected to this first carbon, carbon number 1, you have a nitrogenous base. Okay. Then connected to this fifth carbon, you have a phosphate group that you can indicate by this. Okay. So you can call it the phosphate group. Okay. Quickly draw this in this space. Quickly draw this. Is there a lag? Anyone whose the video is lagging? I don't think it's from my end. I don't think it's from my end. Is it? Send me a response. Is it lagging? Audio, video lagging? Okay. Okay, right. Quickly draw a nucleotide, pentose sugar, nitrogenous base, and the phosphate group. This is the symbol you need to be able to draw in the exam. Okay, this is the sort of the molecular diagram of it. Hi. Okay. Then a nucleotide without a phosphate group is called a nucleoside. Okay. So if you don't have this part from this point, everything on this side, that is called as a nucleoside. So what you can do is you can you can just indicate if you only include what? If you only include the nitrogenous base and the pentose sugar, that is a nucleoside. A nucleoside. Okay. Nitrogenous base is a hexagon. Okay, that's how you indicate. If you could take the resource book, nitrogenous bases are indicated in this boxy shape, right? But if you go into the actual molecular structure which I have given, they are usually hexagonal shape carbon rings. That is in the case of a pyrimidine, right? So if you come to a pyramid purine, which are longer, you can see they have two rings, no? So we use this boxy, this rectangle again, that is how you indicate any type of nitrogenous base, okay? So don't think about that. Okay, right. So keep this. This is a definition also. They can ask you in the structured essay paper, what is a nucleoside kilovolt? It is what? It's a nucleotide without a phosphate group. Just remember some of the example: adenosine, guanosine. Those are the common nucleosides. Okay. Okay, so now we are going to discuss in detail about each of the components, particularly your pentose sugar. Okay, so pentose sugar you can have two types. Okay, highlight what are the two types: deoxyribose. You can have deoxyribose and ribose. Right? You can have deoxyribose and ribose. Okay, so I have given here, right? You can just zoom in. You can't zoom into the tube like me, okay, right? But you can clearly see in deoxyribose. If you look here, there's only a hydrogen. But if you take ribose, you can see there's an OH. Right? So deoxy means one less hydrogen. So you can maybe circle this in a red color. Make I. You can circle this in a red color or something. Indicating that in deoxyribose there is one less oxygen. That's why you call it deoxy. Okay, one hydrogen atom is less. Then moving on to the variety of nitrogenous bases. Okay, there are two major groups of nitrogenous bases. You classify them based on the size. Okay, so you can see the purine. So I have given purines first, right? What can you say about purines? They have two carbon rings. No, listen. Okay, they have two carbon rings. So they are usually larger. No, they are usually larger. So what are the two purines that you need to know? Purines. Okay, keep in mind purines are larger. Purines are larger with two carbon rings. Study right now. Purines are larger. What are the two purines that you need to know? Write down here: adenine and guanine. Okay, adenine we indicate by the letter A. The symbol of adenine is A. Guanine, the symbol is G. Okay. Okay. Then pyrimidines are smaller in size with a single ring. You can see the pyrimidines. There are actually three types of pyrimidines: cytosine, thymine, and uracil. Cytosine, thymine, and uracil. Okay. So you can see, of course, they have only one carbon ring. Then, okay, someone who missed the nitrogen, our nucleotide, quickly draw this. Okay. Then you can write down the three types of pyrimidines. Okay. You can be very careful of the spellings. Okay. You have cytosine indicated by a C, thymine indicated by a T, and uracil indicated by capital U. All these are capital letters. Okay, all these are capital letters. All these are capital letters. Okay. Right. Then phosphate group, nothing much to talk about. You need to know phosphate group has a slightly acidic nature. Please highlight that. Phosphate group has a slightly acidic nature. Please highlight that. Therefore, nucleic acids are called acids because of the acidic nature of the phosphate group. Acidic nature given by the phosphate group. Okay, that's why they are called nucleic acids. Okay. So you will see whenever we are handling nucleic acids, because they are acids, you cannot have them in a basic medium. In molecular biology, we learn about how to extract DNA, how to store DNA, how to break DNA, how to cut DNA into pieces, or right. So when you are handling DNA, you have to keep in mind it is acidic. Therefore, if you put it into certain alkaline medium, the DNA could be damaged. Okay. So usually mix it with a buffer solution. Okay. Just so I told you this is the foundation for all the basic bio knowledge. Okay, so keep in mind these are the phosphate group. Nucleic acids are acidic. Okay, now we move on to the polymerization, no? Because now you know the monomer, no? How the monomer looks like. So now you can move on to how monomers join to form a polymer. The polymer is called what? The nucleic acid. A new type of bond you need to learn here, right? Such as the glycosidic bond in polysaccharides. You have a new bond here, highlight. What is the bond connecting or forming nucleic acid? Phosphodiester bond. Okay, phosphodiester bond, right? So I have given here, how that forms, how this new phosphodiester bond forms. So you can see, I want you to put a label here. If you can highlight in a different color, that is good. Okay, these bonds are what? These bonds are your phosphodiester bonds. Okay, these bonds, the red color bonds are phosphodiester bonds. Okay, so you can draw arrows. They are phosphodiester bonds. Okay, phosphodiester bonds. Got it? So this is how nucleotides join to form a nucleic acid, right? So we'll see how that forms in word. You need to be able to explain. Okay, you need to be able to explain uh, how these phosphodiester bonds form. Okay, so read with me, Kianwa. Condensation between the OH group of the phosphate of one nucleotide with the OH attached to the third carbon of the pentose sugar of the other. Okay, these bonds result in a backbone. Again, we come to a familiar concept of a backbone. What do you mean by a backbone? Is something that is going to be the same, something that is going to be the same throughout this polymer. Got it? If you remember the proteins, the only thing that changed between amino acids was what? That R group, that R group, the alkyl group. So similarly here, there's going to be a chain, a part in this polynucleotide that you are going to form that is going to stay unchanged. That is what I have indicated by highlighting in a blue color. Can you see? By highlighting in a blue color, that's what I have indicated. So you can put a මෙහෙම එකක් මොකද මේ කියන්නේ මෙහෙම එකක් දෙකක් දාලා you can say this is the sugar phosphate backbone. This is called a sugar phosphate backbone. Sugar phosphate backbone. Okay, sugar phosphate backbone. Okay, sorry, sugar phosphate backbone. Because why are we calling it a sugar and a phosphate? Because this is who? The pentose sugar. Then who is this? The phosphate. So you can see I am highlighted by the blue color are the sugars and the phosphates, one after the other. Okay, so I will show you this with the software right now. Okay, so then you will be able to understand it much easier. Okay, right. So don't think about the two structures that are connected right now. Okay, don't think about the two chains. I just want you to focus on this. Just think of the one side of this. This is what we call as the backbone. This is what we call as the backbone, right? Listen carefully, right? So you can see, make out these sort of these balls. Who are those? Maybe what are those? This. Those are the phosphates. Okay, those are the phosphates. Then who is this pentagonal shape? Pentagons again. Oh, sorry. Who are these? Memeva. Those are the phosphate groups. So now you can see, if I zoom in, if I you can look from underneath even, or you can look from this side. Okay, you can see one of the OH groups. Okay, this OH group through its oxygen is connected to the phosphate. Okay, this carbon through its OH, this is going to be an oxygen, it's connected to the phosphate. Got it? So this is what we call as the phosphodiester bond. This is what we call as the phosphodiester bond. Then it it's going to keep on forming, it's going to keep on forming, keep on forming, keep on forming until you have a repeating set, right? So later it is going to be sort of going to fuse together because it is going to be unchanging. No? So it is going to be unchanging. That's why we call it as a backbone. What sort of a backbone is it? It's a sugar phosphate backbone. That is the concept that I want you to have in mind uh, as we are talking about this formation of phosphodiester bond. So you can see, Metin, so I have highlighted here, they are blinking slightly, the phosphodiester bonds. Can you see? The phosphodiester bonds are kind of blinking. They are formed between the phosphate group and the OH group of the next pentose sugar. Okay. I will show you the proper DNA as well. Okay. Once we have discussed the double helix model. All right. Then keep reading, come back here. These bonds result in a backbone with repeating pattern of sugar phosphate bonds. I want you to highlight this phrase, a repeating pattern of sugar phosphate. A repeating pattern of sugar phosphate units. Nucleic acids are linear polymers of nucleotides. Okay, they are linear, they don't branch. Okay, highlight. Like if you can remember glycogen, okay, there are branching places, no? Right? But in DNA, I mean any nucleic acid, there is no branching. It is going to be just a linear chain. Okay. There are two kinds of nucleic acid depending on the type of sugar. You know this, no? Right? If it's deoxyribose sugar, then it is DNA. If the sugar is ribose, it is RNA. Okay. Then this is important. Okay. So DNA contains, what are the nitrogenous bases in DNA? I want you to remember this very carefully. DNA contains adenine and guanine. Those are what? Purines. Those are purines. Then it contains thymine and cytosine. All good. So what do you have in DNA? I want you to write the symbol. A, T, G, C is found in DNA. Then RNA contains what? Adenine තියෙනවා, guanine තියෙනවා, all good. Cytosine තියෙනවා. But instead of thymine, you have uracil in RNA. So write down the symbols of those: adenine, guanine, cytosine, uracil is U. Got it? Yeah, I'll explain. So entire DNA is going to change based on the nitrogenous bases. That's the next topic that we are going to discuss. Okay. The DNA changes because of the nitrogenous bases, not because of the backbone. The backbone is common. That's why we call it as a backbone. Okay. The backbone is going to stay the same throughout the molecule. The DNA one after the other is going to change because of the changing nitrogenous bases that you are going to have. Okay. So let's see. So in this space, I want you to draw the double helix model. For that, we have to discuss. I mean, we have to discuss how DNA is formed. Okay, DNA or nucleic acids formation. Okay. Any questions? Yeah. Any questions? Okay, right. Then let's move on to the double helix model. Okay, that is was I'll explain what is 5 prime and 3 prime. Hold on, right? When we are forming the DNA molecule. Okay, right. Let's see. DNA molecules have two anti-parallel polynucleotide chains that spiral around an imaginary axis, forming a double helix. Okay, got it? This is very important. Anti-parallel polynucleotide chains that spiral around an imaginary axis. I'm going to explain now what you mean by anti-parallel. Okay, listen carefully. To make a polynucleotide, but more than any polynucleotide, if you want to make DNA, you need two deoxyribonucleic acid strands. You need two deoxyribonucleic acid strands. So you have one here, no? You have one here. You need another one. Okay, you need another one. But the thing is, you can understand. Okay, can you see here? This end that is sticking out, the end of this chain that is sticking out, or let's say the cut end that is sticking out, has what carbon? What is the carbon number? If I zoom in, you can see, no? This is carbon number 1. This is carbon number 2. This is carbon number 3. So, what is the carbon that is on the outermost edge or the cut edge? That is the third carbon. So, you call this end, I want you to write down here, මෙහෙම arrow එකක් දාලා, this is called as 3 prime. Remember this symbol 3 prime end. You can write 3 prime end within brackets. 3 prime end. Why do we call it as 3 prime end? Because the outermost carbon in this end is the third carbon. You can see the number, 1, 2, 3, 4, and this is the fifth carbon. Okay, write down 3 prime end. The phosphate group, this is the phosphate group. This is the phosphate group. Okay. Then, what about the other end of this chain? If you move on to this end, you can also see there, listen, you can see there, if you compare the carbons, 1, 2, 3, 4, this is 5. So, in this upper end, what is the outermost carbon number? I mean, a number of the carbon that is sticking out. That is the fifth carbon. No? So, you call this 5 prime end or 5 prime end. Okay. 5 prime end. So, that is something you need to keep in mind. So, can you understand this nucleic acid chain or this polynucleotide? Yes, this polynucleotide has these two ends sticking out, right? So, that is indicating a certain direction. That is indicating a certain direction of how these polynucleotides form. Okay. Give me 1 minute. Okay. I'm getting a call. Okay. Okay, right. As I was saying, you need to remember what are the ends of the DNA molecule that are sticking out. So now you can understand that, right? So this end is called as the 3' end, the lower end, the upper end, since the carbon that is on the outermost end is called the, I mean, there is the fifth carbon, so that is called the 5' end. Okay. So now I want you to explain, I want to explain what do you mean by this anti-parallel, right? What do you mean by this anti-parallel is this, listen carefully. Okay, so I told you you need another strand like this in order to make a DNA molecule. You need another strand like this. So that other strand, the thing is, it is going to be oriented, sort of standing on its head, upside down. Okay, so I won't draw the entire full structure. But if I'm drawing the pentose sugar, I have to draw the pentose sugar kind of like this. Okay, the pentose sugar I have to draw kind of like this. If I number the carbons, this is 1, 2, 3, 4, and mehema there's a fifth carbon. There's a phosphate group here. Okay, and connected to the first carbon, what you have? I'll use the same yellow color. Connected to the first carbon, you have a nitrogenous base. Okay, then connecting with this phosphate group, you have a phosphodiester bond. Through that phosphodiester bond, there is another pentose sugar, that is connecting like this, and that pentose sugar is again connected to another nitrogenous base, and there's another phosphate group connected to the fifth carbon as well. Okay, so this is the other polynucleotide chain that you need to form a DNA molecule. Now listen carefully. Can you see here what is the carbon that is sticking out? Third carbon. So you call this 3' end. You call this the 3' end. And you call this, you can see, this is carbon number 1, 2, 3, 4, and this is the fifth one. So this is the 5' end. Okay, this is the 5' end. Yes, you need to be able to draw the DNA structure, but you don't need to be able to draw this level of DNA structure. You only need to be able to draw our double helix. That's what we are going to actually draw here. But I'm still explaining this concept of anti-parallel. Okay. So now, as I was saying, this DNA molecule, okay, we say it runs from the 5' when the one we have in the tube, it runs from 5' to 3', that is in this way downwards. Okay, it runs from 5' to 3' in this way downwards. So in this one, what is the 5' meant? It is the downwards, no? So this, you can say, this chain runs again in the 5' to 3', but you can see they run in opposite directions. Got it? They run in opposite directions. Therefore, we call this arrangement as anti-parallel. Simple. Okay, you call this arrangement as anti-parallel. You don't need to be able to draw this, and we'll again talk about this when you are doing molecular. But for now, just keep in mind they form in opposite ways. This chain is going to form in downwards direction now, the other chain is going to form in upwards direction. That is why we call the two DNA molecules are anti-parallel. The two DNA molecules are anti-parallel. Give me 1 minute. That is understood. Neither. Why do we call them anti-parallel? Simply, they run in opposite pace. If one is going down, the other one is going up. Got it? Don't overthink it. Okay. Then they spiral around an imaginary axis. So I'll erase this for now. Natta mega you start to obsess about this because you only need to remember the word anti-parallel. These 5 prime, 3 prime ends are not even mentioned right now. You will think about it when we are doing molecular. The brain has matured, and also with the basis of DNA formation, you need to know what enzymes are forming the DNA, and everything should be going in a proper order. So I don't want to cram this knowledge here because I know there are other things that are coming up. We have to talk about enzymes, photosynthesis. So no need to think about this right now exactly. Just remember, when you are forming this DNA molecule, one runs in one direction, the other one runs in the opposite direction. Okay. Then what do we say? Spiral around an imaginary axis forming a double helix. Why do we call it a double helix? Because you have two things that are spiraling around. I mean, two molecules that are spiraling. So it's a double helix. So can you imagine like a this sort of spiral staircases you have seen, right? You have seen spiral staircases. So if I draw this sugar phosphate backbone in blue color, I can draw it like this. The sugar phosphate backbone, I can draw it like this. How many sugar phosphate backbones do you need? There's two, no? There's two molecules. So I can draw it in this way. Okay, this is the backbone, right? The backbone is composed of pentose sugar phosphate, pentose sugar phosphate, pentose sugar phosphate, one after the other. So you can indicate it with just one line. Okay. Then the two sugar phosphate backbones run in opposite directions from each other. Then this arrangement is known as antiparallel. So this is a small definition. They can ask you what is antiparallel. You need to say the two sugar phosphate backbones run in opposite directions. This arrangement is referred to as antiparallel. Please highlight, because they run in opposite directions. So I can just indicate, okay, if this sugar phosphate backbone is forming in this upward direction, it is being forming. This other one is going to form in the downward direction. Got it? That's why you call it as antiparallel. So what about this imaginary axis? You can see this, there's an imaginary central line, no? There's an imaginary central sort of a midpoint that they are wrapping around. Okay, like this. I'll show you again with the software, you will understand that. Okay. Then the two sugar phosphate backbones are on the outside of the helix, and the nitrogenous bases are paired in the interior. Right. Imagine you this sort of a spiral staircase. You have the sort of the supporting parts of the staircase that includes the handrails and also the outer structure of the staircase. You have seen spiral staircases, right? So you have the handrails that are wrapping around like this. What do you have in between? You have the steps, no? The steps of the staircase, right? So that is kind of similar to the nitrogenous bases that you are going to have in the middle. The nitrogenous bases are going to you have in the middle. I'll show you using the software, then you will understand. Okay. Listen, now highlight this, very important. The two strands are held together by hydrogen bonds. Okay. The two strands are simply connected by hydrogen bonds. Okay. Hydrogen bonds between the nitrogenous bases. Highlight that. There is something you need to know then next, that is the base pair rule. It's a rule that is unique to DNA. Okay. Not DNA, but any nucleic acid, there's a rule that is very important. Please highlight this. The base pair rule is that a purine always binds with a specific pyrimidine. A purine always binds with a specific pyrimidine. Okay. So what are the purines that you know? What are the purines that you know? You have adenine, A. Then what is the other purine that you know? Guanine. Okay. So a purine with a pyrimidine. Got it? Adenine and guanine are the purines that you know. Then who are they going to bind with? Pyrimidine. Listen, this is the binding. Adenine is always going to bind with two hydrogen bonds with thymine. Thymine T. Guanine is always going to bind with cytosine with how many hydrogen bonds can you see? Three hydrogen bonds. This is the base pair rule. Okay. So I'll show you using the software now so that you will have a 3D idea about this double helix. Okay. So interestingly, this double helix was actually discovered by our actually a female scientist. Not I mean the technique was actually done by a female scientist called Rosalind Franklin. Okay, she did a technique called X-ray crystallography, which is one of the ways that we can visualize molecules, one of the ways that we can see molecules. So she did the sort of the work, hard work. Then there was two scientists that were called as Watson and Crick, James Watson and Francis Crick, I think. They saw the experiment done by Rosalind Franklin and they sort of interpreted it. They thought, okay, this is the finding of her experiment. Then they sort of gave to the world the theory, okay, the DNA of humans look like this. They look like a double helix wrapping around an imaginary axis, blah, blah, blah. They described. But who discovered? Rosalind Franklin. Okay, but therefore, but since they described it and presented it to the world, it's known as Watson and Crick model. So that is kind of how the scientific world works. Okay, what to do? Right. I'll show you the 3D view. Okay, now look at this. Okay, so where were we? I showed you how the sugar phosphate backbone forms. This is phosphate, sorry, this is sugar phosphate, sugar phosphate. Okay, so now let's come into the center, right? In the center of this helix, what are you going to have? You are going to have nitrogenous bases. So now can anyone tell me how do you identify the purines in this diagram? How do you identify what are the purines? How do you identify the purines? How do you identify what are the purines? How do you identify what are the purines? Yes, if they are bigger with two rings, that they are probably purines, no? So you can see this red one and this blue one, they are kind of bigger, no? So they are probably purines. Then this green one and this yellow one, green one and this yellow one are smaller. They are so probably pyrimidines, no? Smaller. Okay. So what is the base pair rule say? Adenine, I mean base pair rule says a purine binds with the pyrimidine, no? So if you think, can you see there's these dotted lines are the hydrogen bonds. These dotted lines are the hydrogen bonds. So you can see this purine is bonded with this pyrimidine. We don't know by two hydrogen bonds. So you can now identify what is this red color purine? No? What is the red color purine? Yes, struggle can. What is the red color purine? If it binds with the pyrimidine with two hydrogen bonds, it should be adenine. Yes, so අපි බලමු හරිද කියලා. Yes, it is adenine. Can you see? It is adenine. Then, so who is this supposed to be? Who is the yellow one then? Adenine always binds it. Binds it? Thymine. Nah. So this is thymine. No? Got it? Then, of course, the other purine, this blue or purple color purine has to be what? It has to be guanine because it has two rings. And also, can you count 1, 2, 3 hydrogen bonds? So it should be connecting with cytosine. So you can see the green color is the cytosine. So that is what we call as the base pair rule. Now can you see in this DNA molecule, you have the two sugar phosphate backbones running on the outside. In the center, you have nitrogenous bases which are connected by hydrogen bonds. So if you go up, we are going to see they are going to start spiraling around. They are going to start spiraling. Now this is the classical DNA you have seen everywhere, right? The classical DNA molecule. So this is what we call as the double helix. Got it? Because you have two nucleotides that are spiraling around an imaginary axis. Okay. This is a double helix. Anything else I can show you? Not much. It is exactly all the same. These other levels, so how this DNA then coils and packs to form a chromosome is this. Okay, so we'll come. I'll show this again when we are doing cell division. Okay. Yeah. These are called histone proteins. When this DNA is going to wrap around these protein units, these are called histones. Histones ke agastones neve histones. It's a type of protein. Okay, not anyone's stones, but histone protein. Okay, right. DNA is going to wrap around it. So if you zoom out of this DNA, there is a structure. It's a double helix composed of sugar phosphate backbones running in opposite directions with nitrogenous bases in the middle connected by hydrogen bonds. Got it? Simple enough. So what I want you to do is now come here. We are going to draw. We have something more to read. Since the two chains or the strands are complementary to each other, because why are they complementary? Because of this base pair rule. Okay, so these pairs are known as complementary base pairs because they are sort of following a matching sort of they are pairing up according to a rule, no, right? So in this original double helical structure, one complete turn consists of 10 base pairs. Okay, so you can see one turn consists of 10 base pairs. 1, 2, 3, 4, 5, 6, 7, 8. Metana me hidden 9, 10. Okay, one twist contains 10. Okay, one twist contains 10. So just highlight that also. 10 base pairs in a turn. So you can do this as homework, but I want you to draw here a DNA double helix because in 2021 A-level paper essay question, you had to draw the double helix, so that it's very important that you are able to do this. Okay, so we'll draw, this is the sugar phosphate backbone. Okay, this is the sugar phosphate backbone. Got it? A bit ugly, but anyway, then using some nice colors, I want you to show this is guanine. Okay, so guanine is going to bond with what? Guanine is going to bond with cytosine. Then I want you to indicate the number of hydrogen bonds. 3. 3 hydrogen bonds. Okay, then draw adenine, and adenine is going to bind with thymine by how many hydrogen bonds? 2 hydrogen bonds. Okay, so indicate that. Then later you can just show the base pairing by just some of these sort of lines, but separate them at the middle so that you can see where the nitrogenous bases are connecting. Okay, you can draw this, complete it once the class is over, so you can keep some space. Okay, in any diagram the labeling is the most important. Okay, the labeling is the most important, so I want you to label what is this blue color, you can label it as sugar phosphate backbone. Sugar phosphate backbone. Okay, there are some questions. Is there any patterns when the next base pair comes? That is called as genetics, right? the arrangement of base pairs, how the base pairs are arranged, A, T, G, C, G, T, A, C, C, A, G, 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 T, T, C, T, T, C, C, whatever that pattern is completely inherited by from your parents. Okay. We no, you don't need to indicate the pentose sugars or the phosphate groups. You only need to draw a sugar phosphate backbone as a line, but you need to indicate the nitrogenous bases at least, so that you can indicate the base pair rule. Okay. What else? Any other questions? Can nucleoside exist naturally in cells without becoming a nucleotide? Yes. You will see adenosine when we are doing ATP. ATP can break down into a adenosine. Adenosine triphosphate can break down into just adenosine. Very difficult to draw a double helix. Maybe right? Don't think about it. Just something close to it. Okay. Then I want you to, in this space, in this dotted lines, I want you to write down the base pair rule. Okay, adenine binds with 2 hydrogen bonds with thymine. Guanine binds with 3 hydrogen bonds with cytosine. Okay. Then within brackets also write methana 2 H bonds, adenine and thymine 2 H bonds, within brackets here 3 H bonds, 2 H bonds and 3 H bonds. Okay, so once you draw this, do you want a break or do you want me to finish RNA as well? But again, break on it. I don't have a break. Okay, today at least I don't have to go back to hospital, but usually on Mondays I finish at 10 and I go to hospital again. Today I don't have to. Okay. When are you not busy? I don't know. If I. That's the thing, right? When you can work, you have to work. Then you can take a break. When you can't work means you are physically unable to work when you are old. So when you are old, you can't work. Then, since you can't work, you stop working. When you are young, you can work, so you work. What type of a doctor am I going to be? Probably a good one, okay? Hopefully a good one. We'll see afterwards. Okay, right. So that is DNA. Any questions? Functions of DNA, Taggala, let's highlight. Store and transmit genetic information from one generation to the next. Yes, we talk in cell cycle. Okay, how in the mother cell the DNA is copied and it's nicely given to the daughter cells. In mitosis, it is done exactly. In meiosis, it is sort of halved and given into the four daughter cells that are produced. So highlight, store and transmit genetic information from one generation to the next. Then store the genetic information for protein synthesis. Yes. Okay. By looking at the information in DNA, your body is going to make what? Proteins. Okay. Because if you imagine, I use this example, your proteins are is like a cake now. A protein is like a cake. The recipe for the cake is in the DNA. Okay. So you have to take a look at the DNA recipe to make the protein cake. Otherwise, the protein cake is going to be messy. Okay. Not Lionel Messi, but it's different. Okay. So the recipe for proteins or the information on how to make a protein is in the DNA. So those are the two functions of DNA that you need to know right now. Okay. So then we have to move on to RNA. You can take a small break. Okay. Afterwards, we'll quickly finish our types of RNA. Then we have to do a comparison, and this tute will be over. Then we are going to move on to microscopes. Okay. So we have a lot of work remaining to do. Taggalend. Okay. Small 10-minute break. 10 minutes. Come back quickly. Okay. Right. By 8:12. 8:12 at the come back. Right. Right. You can ask any question. You can send to the chat and I'll answer. Okay. Okay. Right. Can you hear me? Okay, break is over. So, do you want to know how you do? Have you heard of something called putting on new lenses? අලුතෙන් lens එකක් දානවා එහෙම නැත්නම් like if you have grandparents or something, have you heard of something called putting on a new lens to your eye? Have you heard of something like that? How do you put a new lens to your eye? අහලා තියෙනවද? Yes. Cataract surgery කියලා right? When you have the lens of your eye is something like this. Okay, right? The lens of your eye is something like this. You know the lens, no? It is how you bend light waves. Okay, it is actually a jelly like substance, transparent gel that is in this sort of a capsule. Okay, I'm talking about the eye here, don't be confused, I'm just saying something extra. Okay, so it has sort of a casing, and inside that casing you have a gel that is actually the proper lens. So this is the totally the lens. So what's going to happen is when you get a light ray, this is going to be bent like this, right? This is what happens in the lens. But when you get older, this the central part of this lens, this jelly like thing, can become cloudy, and sort of it becomes less transparent. Okay, it becomes cloudy, so that light rays don't go through it very effective. Got it? So that's what happens. So when a person, we say a person has to undergo a cataract surgery, මම අලුතෙන් lens එකක් දාන්න ඕනේ කිව්වහම, that's what's going to happen. We are going to take out or suck out this middle gel like now with dirty gel like material, and while keeping this outside capsule undamaged, then we are going to put a new artificial gel like material inside. Do you want to see how that works? Okay, so it is kind of like this. So we were able to see this during our ophthalmology appointment. Okay, so we can just take a look at this. Okay, so ophthalmology is a very interesting field. You require a lot of knowledge as well as you require a lot of practice. If you are very good with your fine skills, then you can try to be a eye surgeon. Okay, what we are trying to do, this is all done using a microscope. The surgeon is looking through a microscope, and this is the eye. Okay, some people can't watch things being done to eyes, I know. Okay, right. Listen, so can you see all these are microscopic and it's kind of robotic, okay? Very small scale, and the eye has been numbed, okay? So what we are doing actually here is we are breaking into pieces the old that dirty lens, okay? We are breaking into pieces that old dirty lens into pieces, okay? Then we are going to suck it out, okay? Yes, you can see it's being broken into pieces, right? You have now torn it up, and we are going to break it into more and more pieces. This is not inside of your eye, Harida. This is just on the surface. That is just the lens that is in the front of your eye. Okay, got it? So the surgeon is looking through a microscope, and you can see he is breaking the old lens into pieces. Okay, then what he's going to do is he's going to suck it out. Okay, now this is the new lens. And again, you see, you will be able to see the new lens is going to be held by this small let's say handling tool, and it's actually folded into half. You like a circular piece of paper, you fold it into half, right? Then what you do is you, now the lens, inside of the lens is clean, you took out all the dirty old lens. Now what you're going to do is you are going to put in the new lens. It is been folded into two because otherwise you can't put it inside, no? Then the surgeon is going to nicely, using the tools, he's going to just adjust it inside the eye, so that Arthin, can you see the transparent glass like thing? That is the brand new lens that is going to go in. Got it? And now he is finally adjusting the new lens. Can you see how transparent it is now? That is the new lens he is adjusting, right? So that it fits nicely. Can you see the edges of it? That is the new lens. Okay, it's playing again from the beginning. Okay, if you jump to the beginning, you can see how earlier this was the old lens. Can you see how cloudy it is? Like it's dirty, no? That is what we call as having a cataract. Okay. So that's what happens in when you say you are a grandparents or when they have gotten a cataract surgery. අලුතෙන් lens එකක් දැම්ම කියලා කියන්නේ. Right. That is this. Okay. The synthetic lens. The synthetic lens is going to be placed very carefully like this. Usually there are German lenses, American branded lenses. So many lenses are there. Right. So now see, can you see it's very transparent, no? Very clear now compared to earlier. So the person can see clearly without being, it's not like looking through a misted glass, frosted glass. Now very clear vision. Okay. Good enough. All this is done by looking at through a microscope. The surgeon is looking through a microscope. Okay, it's very small. Got it? You need to have a lot of eye control, hand-eye coordination. Okay, in order to become eye surgeon. And these instruments are very, very expensive. Got it? Okay, okay. Come back to your RNA. Come back to RNA. Right. So we have finished the discussion about DNA. Next up, we are going to talk about ribonucleic acids. Okay, ribonucleic acids. Okay, page number 21, structure of RNA. It's normally a single-stranded molecule, single-stranded nucleic acid, highlight usually single-stranded, composed of ribonucleotides. Because why? What is the pentose sugar here? What is the pentose sugar? It is ribose. No, you can write here ribose. What are the bases? Highlight uracil. But instead of a uracil, instead of again, you have uracil in RNA instead of thymine. Can we get rid of wearing glasses? Depends on what sort of a weakness you have. There are laser surgeries to correct some of the issues. Okay. Okay. So highlight what are the bases: cytosine, guanine, and adenine. Okay. Complementary base pairing between two RNA molecules or within the same molecule may occur, right? So compared to DNA, in RNA, not always there's going to be complementary base pairing. So sometimes it can exist as a single molecule, but sometimes, rarely, two RNA molecules can join, or within the same molecule you can have. For example, if you take your transfer RNA, tRNA, can you see this is the same RNA molecule? So this is the same RNA molecule that is going homo gilla. This is the same RNA molecule. Got it? But in the same RNA molecule, you can have, so in some places, this sort of hydrogen bonding. Right? You can have some hydrogen bonding. That is called as, within the same molecule you can have complementary base pairing. So tRNA is an example for that. We'll come there. Okay. Complementary base pairing facilitates the 3D shapes essential for their functioning. You need to know. They can ask you why in RNA, what is the purpose of complementary base pairing. If you don't have the complementary base pairing, they can't be properly functional. Okay. Because of the 3D, because of their 3D shape, they can work as working molecules. Got it? Adenine binds with uracil with 2 hydrogen bonds, and guanine binds with cytosine with 3 hydrogen bonds. Okay, so there's a base pair rule here also. There's a base pair rule. I want you to write down the base pair rule. Adenine, you don't have thymine with 2 hydrogen bonds, uracil, guanine and cytosine, no issue. Quickly write it down, the modified base pair rule for RNA. Okay, that is a modified base pair rule. Then we'll move on to individual types of RNA that we need to discuss. Okay, to understand RNA as a whole, remember RNAs are the main molecules involved in making proteins. So, in order to understand the functions of RNA, you need to understand how proteins are made. But I'm not going to tell you the full story, but I'm just going to tell you a short summarized version of it so that you can understand the main roles of RNA here. But later, when you are doing molecular biology, we'll discuss the full story. This is now like a trailer of the full movie of protein making. Okay, right? Listen, listen carefully. This diagram is going to summarize the how you make a protein in your body. I'm going to explain it using an example as well. Got it? Imagine there's a library containing recipes. Okay, so you are planning to make a cake. The cake is a protein. Okay, so but to make the protein, you need the to make the cake, you need the recipe. The recipe is in a library, but you can't take the recipe out of the library. Okay, so the library is your nucleus. Okay, the library is your nucleus. What is the recipe inside? The recipe is what we call as the DNA. DNA contains how to make the protein, the information on how to make the protein. But you can't take DNA out. Why? You are going to make the protein at a different location, or you are going to make the cake at a different place that is called the ribosome, where ribosome is the place of protein synthesis. Okay, ribosomes, you know. Then, so what's the problem? You can't take the recipe to the kitchen to make the cake, right? So from the nucleus, you can't take the DNA out towards the ribosome. You need the information in the DNA. What you are going to do is you are going to go to the library. You are going to take a photocopy of the recipe. You are going to make a photocopy, or you take a photo of the recipe book. That photo of the recipe book that you take, that you can take out. You can photocopy the books in the library, then you can take out the photocopy. Similarly, since you can't take out the DNA, what you do is you make a similar copy that is a type of RNA that is called mRNA or messenger RNA. The messenger RNA is a copy of DNA, or it's a messenger. This messenger RNA can come out of these pores in the nuclear envelope that we'll discuss today. You can write here, these are called nucleopores. Nucleopores. Then that mRNA is going to come out. What is this mRNA now? It's the photocopy of the recipe to make the protein. Then you bring it to the kitchen. Then in the kitchen, by looking at the mRNA recipe, you are going to make a protein. You can see this is the polypeptide chain of the brand new enzyme, sorry, brand new protein being made. Okay, so that is how you make proteins in the kitchen. To help you make the protein or the cake, so imagine your mother is making a cake, you are the mother's servants, right? Because why? If you and your siblings, one has to bring the sugar, one has to bring the eggs, one has to break the eggs, other has to bring the flour, and everyone has a role. So at the ribosome also, when you are making proteins, you need amino acids one after the other. Okay, you need to bring amino acids to the ribosome. So in order to do that, there's a servant. This servant is this. This is our tRNA. tRNA is the servant who helps the making of protein. He brings amino acids towards the ribosome. Got it? He brings amino acids towards the ribosome. That is the basic simple explanation of how you make protein. Okay, right. So we'll go through the individual RNAs. Read. You need to remember the functions. Okay, significant features. Messenger RNA is a linear molecule. Within brackets, can you write here mRNA? mRNA, simple M, the rest is capital. mRNA is a linear molecule and least abundant. You don't have much mRNA. It has two functions. Copies the genetic information stored in DNA molecule as a sequence. Copies the genetic in DNA as a sequence of nitrogenous bases. So you can write here. So what do? You convert DNA information in DNA into RNA. Okay. That process has a name. Okay. Just write here the name, we'll talk about it later. That is called as transcription. Transcription. Okay, transcription. That means you are making a photocopy of the DNA. What is the photocopy? Is the mRNA. That process is called transcription. DNA into actually mRNA is called transcription. 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 Then next function, transport genetic information from nucleoplasm. Can you correct this? There's a small printing mistake here. Plasm, මෙතන ඒකක් නැහැ. Plasm. Nucleoplasm to the site of protein synthesis through the nucleopause. Highlight. Nucleoplasm means this inside of the nucleus. It is like the cytoplasm of a cell. From the nucleoplasm through the nucleopause, where do you bring? To the ribosome. Ribosome is the kitchen, protein making kitchen. Okay. So that is mRNA. Then tRNA or the transfer RNA. Okay, highlight. Smallest RNA, linear, but forms a 3-loop structure. Very important. This is a very unique structure of tRNA. අපි එකට කියනවා 3-leafed clover එකක් කියලා. Clover leaves you have heard. Don't think about it. It's a 3-loop structure. 1, 2, 3, right? Function, transport amino acids to the site of protein synthesis. So here, like for example, you are helping your mother to make a cake. Okay, you bring the flour, right? Flour means piti from the by holding it with your hand. So the tRNA has a place to carry the amino acid as well. That is called the amino acid attachment site. So it can carry the amino acid and bring it to the ribosome. Okay. Ribosomal RNA next. Okay. Ribosomal RNA, highlight, it is the most abundant type of RNA because why? You have ribosomes everywhere in the cell. You have ribosomes everywhere in the cell. Therefore, it is very abundant. rRNA has a complex irregular structure, highlight. You don't need to know the structure. It's a complex irregular structure. It is a site where polypeptide chains are assembled or the protein making site. Okay. Protein making site. Right. Then, so understood. Got it? As homework, we are going to write a short note. Okay. We are going to write a short note. Please write down in your essay writing book. Please write down in your essay writing book. Short note. For the first time, we are going to write a short note. Okay. For the first time, we are going to write a short note. Okay. Listen. How to write a short note is this. Give an attempt. In a short note, you do not need to write full sentences. You can write in point form, but do not put bullets or stars or dots or whatever. Right? Write one after the other as sentences, or you can, one after the other in the rules, but you don't need to write in full sentences. For example, right? So you can say, okay, mRNA is a linear molecule and is the least abundant type of RNA. It copies the genetic information in DNA, right, as a sequence of nitrogenous bases. So of course you can write as sentences, but you don't need to. Okay, then you don't have to say it transports, or you don't have to say mRNA transports. You can just say transport genetic information from nucleoplasm to the site of protein synthesis through nucleopause. So you can write as point form in a short note. Okay, you can write in point form. Okay, the short note topic, write down. So you know how is the short note question given? The short note question is the final essay question in your essay paper. Okay, take a look at the essay paper. I'll show you an essay paper. Listen carefully now. Passe don't understand kiyanne epa. Okay, right? If you take a look at an essay paper, have you at least checked the A level biology paper? If you go to the essay paper, මෙන්න මේ වගේ තියෙන්නේ. Right? This is how you get the essay paper. No? Right? In the essay paper, the 10th question, right? Out of these five, you need to write four. No? 1, 2, 3, 4, 5, sorry, 6. Out of these 6 questions, you need to do only 4. You can remove, you cannot do 2 questions. Okay? So you can either write the short note or not. Right? It's up to you. But if you write the short note, you have to actually answer 3 questions. For example, Metin, artificial breeding in plants, Okazaki fragments, and desert plants. So they are the topics that they have given. So you can see, in order to get the full marks, you need to write all 3. There are 3 topics, right? There are some advantages and disadvantages of doing the essay question, sorry, short note question. One thing, because short notes question you can write in point form, sometimes you can save a bit of time. And the other thing, if you take an essay question, for example, then if you take an essay like this, process of secondary growth, if you don't know the secondary growth, you can't answer that. No? Nothing you can write about the essay. But imagine you don't know about desert plants, but you know these two. So imagine you have written 3 good essays, but you need another 1 more to write. Okay. Then a short note is a place where you have 3 topics. So if you know about 2 topics out of 3, you can do this. So that is the advantage. One thing, you can save a bit of time. The other thing is, because there are 3 topics to write, you can still gather some marks. Okay. The disadvantage is the opposite of those. Sometimes, because you have to write 3 topics, sometimes the short note can take a bit longer than you expect. So it is up to you to strategically decide, okay, if I'm going to write the short note or not. Okay. So listen, the short note topic I'm giving you today is only 1/3 of a short note question. So I want you to write structure and function of RNA. Structure and functions of RNA. Structure and functions of RNA. Write down. No, you can't draw tables or anything. You have to write one after the other. Okay. You can write in point form. You can't draw tables. You can't put star marks, those kind of things. Okay. I think I wrote in my A level year, I wrote the short note. I think so. Yes. Yeah. Because I was running out of time. You will understand, from coming from the structured essay paper into the essay paper, you are going to have a time issue in the second part of the paper. MCQ again, you will actually have a lot of extra time. But in the second paper, time management is going to be a small issue. Okay. Structure and functions of RNA is the essay topic for this week. Essay writing book. Everyone has bought an essay writing book at this point, right? Yes. At the end of these 2 years, you are going to have a nice collection of essays that you can be satisfied. Okay, before the day before the exam or something, you can just take out your essay writing book and see, okay, I have worked hard. I have written so many essays. Okay, so I'll be okay tomorrow. You can't number the points. Don't number the points. No need to put bullets, dots, whatever. Just write after one after the other. Okay. Don't overthink this. Or don't put the numbers. You can leave a rule. Yes, you can leave rules if you feel like that is going to make the answer more clear, so that the examiner can see your writing more clearly. Then you can leave a rule. Yes, you can in the examination hall you can get as much as papers you want. Okay, so that won't be an issue. You can leave rules if you feel like it's going to make it much neater. Right. Okay. Final part. We'll complete this. Differences between DNA and RNA. Differences between DNA and RNA. Okay. We'll write on this side DNA now. On this side RNA. What are the differences you can see? Tuggle again. What are the structural differences? DNA is double stranded. DNA is double stranded. RNA is always single stranded. Only one RNA gets involved. Okay. Even if you take something like tRNA, you can see it is still the same molecule. It is still the same molecule. No two molecules. I mean, there are no two molecules. Okay. Then DNA has adenine, thymine, guanine, cytosine. RNA has adenine, thymine, guanine, uracil. I'm sorry, adenine, uracil, guanine, cytosine. Adenine, uracil, guanine, cytosine. Then you can write sugar in DNA is deoxyribose. Sugar in RNA is ribose. Sugar in RNA is ribose. Did I give a homework last week? Are diagrams needed for the RNA assay? Yes, I want you to draw the tRNA. Okay, I want you to draw the tRNA molecule. Okay, I want you to draw the tRNA molecule. Yes, actually, since I gave it to you, I mean, we won't be practicing to draw it here, but I want you to write in the. I want you to draw that when you are writing the short note. Okay, so got it. You need to draw a diagram of tRNA under tRNA. Okay. Do the labeling as well. Amino acid binding attachment site. Okay. Clear. So with that, we'll come to the final part. Okay. With this lesson. Okay. Let's. The tRNA I have given, yes, you can draw that, but it's a bit complicated with this mRNA also involved. So you can just draw the diagram in the resource. Okay. You can draw the diagram in the resource. Got it? And also no need to draw this no need to draw this thing. This diagram you don't need to draw. Only you need to draw the 3-loop structure. You only need to be able to draw this 3-loop structure. Got it? No need to draw that complicated. That is actually how the real mtRNA looks like. This is how the real tRNA looks like. But you know, in biology, we don't draw the real molecular structures. No, we only draw symbolically. We only draw symbolically. How to know which diagram should be drawn? That comes with practice, and also the most of the simple diagrams that you have in the resource book, you should be able to draw. You should be able to draw in the exam. Okay, I'll mention to you what are the diagrams you need to practice. Okay, then we can practice in the class by writing essays. Okay. Okay, then we'll move on to the other types of nucleotides. There was a question, are there any other nucleotides other than deoxyribonucleotide and ribonucleotide? Yes, there are. Yes, one thing is ATP. We did this in theory class. What is the long name of ATP? You can write in here, adenosine triphosphate. Adenosine triphosphate, ATP. Then universal energy carrier, it is the energy currency. Okay, it is a high energy molecule that you can use to do any energy demanding reaction inside the cell. Okay, then NAD plus, we'll soon learn about NAD plus in the theory class. Okay, that is under cellular respiration. Yes, Medine, you can highlight. Acts as a oxidizing agent during respiration. Please highlight that. And also coenzyme electron carrier. Then you already know what is NADP plus, right? If you joined last week in the Saturday group people, now you know how NADP plus is being used in the Kelvin cycle. But everybody knows how we produce NADP plus. What do you make from NADP plus? You combine it with hydrogen to produce what? NADPH. You know this, no? In the light reaction. So it is acting as a S. What is it acting as? It is able to, so it's an electron carrier, right? It's an electron carrier. Therefore, it is going to act as actually an oxidizing agent. Methana, I'm very sorry, there's a small mistake here. Can you please correct this? It acts as a oxidizing agent, okay? It acts as an oxidizing agent, because can you understand, okay, how do you make NADPH? I hope you can remember from last week from the light reaction, there's an enzyme, no? NADP plus electrons plus H plus, you are going to produce NADPH. So this NADPH is able to convert back to NADP plus by releasing its electrons. Okay, so by releasing its electrons, so when those electrons are released, okay, what's going to happen? That means, so in here, what is NADP plus doing? He's accepting electrons, no? In here, he is accepting electrons. So somebody else is giving the electrons. Somebody else is giving the electrons means that that person is undergoing oxidization. So this guy is oxidizing the other person. No? NADP plus is oxidizing the other person. Okay, that's why we call it as an oxidizing agent. So even NAD plus is an oxidizing agent. Okay, please correct it there. Cut it and correct it as oxidizing agent in photosynthesis. Then there's another one, FAD, flavin adenine dinucleotide. Okay, that is also an electron carrier and also coenzyme. Okay, those will be questioned in MCQs usually. Any questions? Okay, we'll do one MCQ. So with that, this duty is over. Okay, you can do this one. Some nucleotides, quickly do that one. So listen, as you are doing the question, now we have completed, actually the chemical basis is now over. Next, you have to watch my practical seminar video. Okay, you have to watch my practical seminar video. Listen carefully. There, it has two parts. The practical seminar had two parts. One is the identification. One is the identification of biological molecules, that is reducing sugars, non-reducing sugars, proteins, lipids, and starch. That is part one. Second is identification of the identification of the parts and using the microscope, light microscope, to observe an onion peel under the light microscope. So in our practical seminar video, you have two parts. I want you to watch that video completely. Got it? If you can't watch it at once, you can watch it in several times. You can speed it up or whatever. Okay. So I'll send it to all the groups. You can watch it. Got it? And you have to complete the tute. The tute is going to be the second tute that you might have received. Out of the pair of second tutes that you received, this one, you received a question practical guideline, right? So in there, if you turn from page number 12 onwards, it is all about our practicals. So in the practical seminar video, I tell you how to complete all these empty spaces, including parts of identification of parts and functions of light microscope and using light microscope to observe specimens. That also I explain, and you have to draw a microscope here. Okay, up to there you are going to be able to complete using the practical seminar video. Listen, okay, that is very important. So the next thing for us to do is to move to our second tute. There we have to start with microscopes. Okay, so all of you have gotten this tute, right? Anyone who doesn't have this, hands up. Hands up who doesn't have this tute. Yeah, I mean, if you have enrolled already with the payment, it would be delivered as soon as possible. Okay, I apologize for any delay, but I'll look into it. Okay. Do you have the PDF? Have you received this tute? Did you receive this one, that the one we are doing right now? If you are, I mean, for brand new people, all the three tutes are coming as one package. Okay. Right. Okay. Tell me, Taggala, what is the answer for this MCQ? What is the answer? Some nucleotides contain hexose sugars. Yes. No, it's always all pentose. Neither. Act as organic cofactors. What do you call as organic cofactors? We learned about cofactors when you are doing enzymes. Okay. Uh, what do you know? What do you call as organic cofactors? That is, cofactors are sort of molecules that are essential for certain enzymes to do their work. Okay, so some enzymes can't work without these cofactors. So actually, organic cofactors have another name. You can write here. They are coenzymes. Organic cofactors has a special name as coenzymes. So yes, that is the answer. Some nucleotides act as organic cofactors. What are the nucleotides acting as cofactor? Organic cofactors you can write down NAD plus, NADP plus, and FAD. They don't serve as enzymes, they only serve as coenzymes. Do they act as electron carriers? No, you can correct. Sorry, do they act as oxygen carriers? No, you can correct it as they act as electron carriers. Do they serve as a food reserve? No, you don't usually use nucleotide energy as food. Okay. Any questions? Okay. Let's move on to microscopes. Any questions? Yeah. So actually, if you are going to watch the, I mean, I want you to watch the practical seminar video. When you are watching the practical seminar video, there I actually explain the whole working process of microscopes very in detail as well. But today I'll quickly give you a brief overview, okay, so that you understand the basics before you watch the practical seminar video as well. Okay, so what I'm going to do is I'll read the resource book together with you, and we'll talk about electron microscopes, those kind of things as well. So listen carefully, we have to cover a lot, therefore I want your full attention, Haridha, right? So you can understand compared to the normal theory class, I'm going a bit faster, and I'm sort of giving you, I'm expecting you to work faster as well. That is the thing what to do. We have to be up to speed, right? Very fast. Okay, right. So take out the our second tute. Page number 1. Anyone who doesn't even have the PDF, I'll send the PDF to the group. Sorry, Zoom chat. It will be delivered as soon as possible. Okay, I know it needs some time for you to get used to this website enrollment process. Okay, it was difficult for us because from our times we started this whole online learning thing. You were learning with online platforms since your O levels, right? We had to adapt into O level. Sorry, this online uh, learning sort of midway in our A levels. Okay, so we had to go through those difficulties. You guys are now probably very used to this. Okay. Okay, right. Microscopes. Come to the resource book, page number 22. Contribution of microscope. So you can understand now, okay, you will immediately see a difference between our first tute and the second tute, because our second tute doesn't have any resource book text. The second tute doesn't have any resource book text. It's only diagrams. Because at this point, one thing you need to have resource book as a printout with you, and the second thing, I can't actually give you the resource book printed out. If someone is doing that, that is illegal in a tuition class as well, right? Because it's we are not supposed to sell it. Okay, I'm charging you a fee, no? So I give the resource book in the first tute because many of the students in the early days, they don't have the resource book. So that's why I send it. I mean, I included in my tute, but afterwards I am not going to give you. Okay, you need the PDF on. I think I send to every group. People, are you not in the groups? I think everyone should be in our online groups at this point. Okay, there's a notification group as well as there's a group 1 and group 2. Okay, so for physical people, there's a ISM group, there's a headway group. Okay, I just now send to the Zoom chat as well. Okay, got it. If you're not in a group, please get added to a group. Send me your number so that I can add you to groups. Okay. Okay, right. We'll come to microscopes. Let's keep reading the resource. Okay, okay. Now quickly, can you download it from the Zoom chat? Okay. Come to the resource. Okay. Advancement of cytology is mostly based on microscope. Okay, discovery and early study of cells progressed with the invention of microscope. Right. What do you mean by cytology? What do you mean by cytology? It's a study of cells. No? Cytology is a study of cells. Great. We talked about it in introduction to biology, right? Cytology, it's actually another name for what? Cell biology. Okay. Okay, light microscope. Visible light is passed through the specimen and then through glass lenses. The lenses reflect light in a way that the image of the specimen is magnified and it is projected into the eye. The simplest microscope is a hand lens. So highlight those things. Okay. Simplest microscope is a hand lens. They can ask you that sometimes. Structured essay paper. So you can see why we had to invent the microscope, right? So this is the scale of our certain. Can you see how long a new nerve cell is? Right? Can you see how long a nerve cell is? A nerve cell is long up to 1 meter in length. Because can you see from the sort of beginning of your leg, from the spinal cord to the end of your leg, a single nerve can go. The nerves get bundled together. So the individual nerve cells get bundled together to form a very long nerve. So to your arm, from your spinal cord, nerves are going along the entire length. It is only, it's one cell, got it? One very long, very thin nerve fiber. So they are very long. Okay. Then you can see the scale that is going. So you can see most plant and animal cells are at around 100 meter to 10 micro, sorry, 100 micrometer to 10 micrometer range. Then you move on to the organelles. Then you can see where light microscopy begins and ends. Where the light microscopy begins and ends. Okay. Then super resolution microscopy is sort of advanced light microscopy. We can do something called oil immersion. There are some techniques. Okay. But the light microscopy is going to stop at kind of the level of the smallest bacteria, that level. Then we have the latest, not latest technology, it's now a very old technology, electron microscopy. Electron microscopy, you can go almost up to the level of molecules, up to 1 nanometer maybe in size. Then you can't observe atoms. Even with electron microscopes, you need certain other techniques such as X-ray crystallography and other kind of things to see atoms. So you can see this is how our microscopes progressed. Okay, that's what we are going to learn. Got it? So mainly we are going to learn about the compound light microscope first. Then I have given you a compound versus electron microscope comparison. Then electron microscopes, there are two types. That comparison we are going to do. Okay. Any questions? So come back to the resource. Compound light microscopes are commonly used in school labs. It is and used in medical laboratories as a diagnostic tool. Okay. So you can observe so many things under the microscope. You can detect cancer. You can detect so many disorders and other abnormal cells when you observe them under the light microscope. Okay. Okay. Keep reading. Resolution, power, and magnification are important parameters which can be seen in a microscope. Okay. What are the two parts of a microscope that you need to know? What are the two parts of a microscope that you need to know? So in physics, you will actually draw these. Okay, you will actually draw the microscope, and also you should be able to draw different ray diagrams in the microscope. There is several adjustments that you can do, and in physics, you have to be able to draw them by practice. Okay, so in bio, what we are going to mainly do is we are going to identify the parts and also see how the microscopes work overall. Okay, so if you take the usual compound light microscope, it has two lenses. Okay, it has two lenses. What are the two lenses? There's an objective lens and an eyepiece. I'll do the labeling on top. Okay, there's an objective lens and an eyepiece lens. We call it the objective lens because the object or whatever you are observing is going to be sort of close to it. Okay, imagine the object is something like this. We indicate objects using this sort of arrow like that's a symbol for the object. Okay, then where do you keep your eye? You keep your eye near the eyepiece. Right, you keep your eye near the eyepiece. Okay, this is the eye. So the light rays from the object is going to travel and it is going to be magnified by the microscope and you will be able to see it. Okay, so the light travels into the objective lens first. Listen carefully. Light goes into the objective lens first, then it will be bent. Bend now, because why? It is refraction, right? Refraction. Then we'll take another light ray and it sort of bends like that also. And they are going to meet at a certain point. You will learn all this very nicely in physics. Okay. Then there's an image that is formed, and the waves from that one will also be magnified further for the second time to produce a very big image, by your second lens, that is called the eyepiece. So this lens, so this ray diagram might look a bit confusing to you, but after you have done it in physics, it will make a lot of sense. Okay. This is basically a rough wave diagram. You don't need to know you don't need to be able to draw accurately because this is bio. Okay. So light rays are going to travel like this and reach the eye. So you can label this, Haritha, you can label this. This is the object, this is the image. Okay, that is the magnified. Can you see, compared to the image, compared to the object, how big the image is? That's what we are trying to do with a microscope. We are going to generate a much bigger image so that our visible eye can see this. Okay. So this ray diagram you need to be able to draw, but I'll fill out everything that you need to write in this space. So you can see there's some space, no space again. I want you to write down some things. I'll complete it, then you can write down afterwards. Okay, right. Then come to the resource book again. Come to the resource book. Magnification is a ratio of an object's image size to its actual size. Okay. So that's a definition. Please highlight it when I tell you to highlight in the resource book. Okay, highlight it. Magnification, write down. Okay, okay, you need to write down. It's the ratio don't write it. Okay, I'll tell you to write. I'll give you some time to write as well. Okay, unknown magnification definition. Okay, magnification is the ratio, okay, of an object's image size divided by actual size. Or you can say object size. This is how you take magnification. So you can see this is the object, this is the image. You can see how bigger than the real object the image is. How bigger than the real object the image is. Okay, so that is the definition of magnification. Okay, then there's another parameter that you need to know. There's another parameter that is called resolution power. Resolution power. So you have seen, right, the resolution of a TV, resolution of a YouTube video. You know what do you mean by the resolution? It's how clear the image is, right? You can increase the resolution. When you increase the resolution of a TV or a YouTube video, or if you are watching this recording from my YouTube unlisted videos, okay, you will be able to see, you can change the quality, right? So the resolution power of a microscope is a measurement of clarity. Okay, it's a measurement of clarity. So the definition is, okay, you can write down minimum distance between 2 points, minimum distance between 2 points that can be, it's given in the resource, okay, distinguished, that can be distinguished as separate points. Okay, write down. It's a minimum distance between 2 points that can be distinguished as separate points. Then keep reading the resource. Keep reading the resource. So I am, because I'm writing a bit big, I'm running out of space, so I'm going to write here. But you can try to keep it in this space. Okay, please try to write in that space. Okay, so keep reading. Come to the next page of the resource. Magnification is limited due to resolution. Okay, so you need to understand. Okay, in a microscope, if you want a clear image, if you want a very clear image, you need to use a type of wave that has a short wavelength. Okay, what do you mean by a wavelength? So if you take a wave that is traveling like this is the wavelength. This distance between this section is one wavelength. If you want to have a very clear image, if you want to have very clear image, it's better to have something with this sort of wavelength, shorter one. Can you understand this? Because that is going to make a much clearer image. That is going to make a much clearer image. Okay, so we have a law that says if you want to have a high resolution power, you need to reduce the wavelength. If you want to have a high resolution power, you need to reduce the wavelength. So you can say resolution power increases when you can. Resolution power increases can when you. What's happened to the chat? Boring. Resolution power increases when you decrease the wavelength. Yes, so it is something that increases when the other thing decreases. No, so you can say it's an inverse proportion. O level maths, do you know this, right? Inverse proportion to what? Wavelength is indicated by lambda, or you can say, you can write inverse wavelength. Resolution power is inversely proportional. You can write it in words here. Resolution power is inversely proportional to wavelength. Resolution power is inversely proportional to wavelength. Okay, so these are the things you need to write in this first page. Okay, quickly take this down. Quickly take this down in the first page. This is all about the light microscope. Okay, quickly take this down, otherwise you're going to forget. Draw a rough diagram, draw a rough ray diagram. There's an object and there's going to be two lenses as objective lens and eyepiece lens. You are going to keep your eye here and you're going to generate a massive image which is called the magnified image. Okay, then write down what is magnification. It is the ratio of image size to the object size. Then write down what is the resolution power. It is the minimum distance between 2 points that can be distinguished as separate points. If you want me to explain that further, for example, if I zoom in, now listen carefully, I'm going to explain what is resolution power. Okay, so I'm going to draw 2 points here. Okay, I'm going to draw 2 points. Can you identify the 2 points? Can you identify these 2 points? You can see there are 2 points here, no? Yes, you can see there are 2 points. Now what I'm going to do is, I'm going to zoom out. Now from your camera, from your home, can you see, I mean, sometimes you might be able to see, if your internet connection is very good, there are two dots here, but it kind of seems like there's only one dot, no? It kind of seems like there's only one dot. You see two dots, then you have a good internet connection. I will draw two dots much close together. Right. Right. Now can you see there are two dots? You can see two dots. Huh? Now can you see two dots? Even I can't see in my own display. That is what we call as resolution power. That is what is called as resolution power, right? So if you have a high resolution power, that means, okay, if you have a high resolution power, that means you can clearly distinguish between 2 points when you have zoomed in maximum. I mean, you are seeing something very big. So in order to improve your resolution power or to improve your clarity, what do you need to do? You need to decrease the wavelength. You need to decrease the wavelength. Okay. Don't lie, you can't see. Got it? Like, I'm trying to explain something. You guys are saying I can see. Look. So this is this I explain this in my the practical seminar video as well. So right now I'm sending the practical seminar video. Okay. So if you have time tonight itself, take a look, because you have the fresh knowledge now of the carbohydrates. Okay. Okay. Sorry. Yeah. Sorry about that. You guys have good resolution power, so you can see the small things, right? No need to draw a very neat ray diagram. I just want you to take a look at the small object and the bigger image size. Okay. Okay. Right. Then, if you take a compound light microscope that you have in your school, right, it is going to have, okay, you're done drawing, right? Okay. I'll send the recording as well so you can complete it. Okay, so you can see, right? I have no other option. We have to go faster. Okay, if you take a school light microscope, something like this. Okay, it's a just a very rough sketch. Okay, so you can see this is the objective lens. You keep your onion peel or whatever here. Okay, then this is the objective lens. There's a lamp here, could be if you can plug it into a socket, it could be a light bulb here. Then from the light bulb there's a light rays coming in, then there's a condenser where you can sort of focus the light rays, where you can sort of focus the light rays into the specimen like this nicely. Then it goes through the objective lens and it will be again magnified. Maybe ocular lens you can cut it and say, in your syllabus we call it eyepiece lens, no? We call it eyepiece lens. Okay, then you can observe it through your eyes, right? So in every light microscope, on the side of these objective lenses there's a label indicating how powerful they are. It could be 100 times. This lens could be 40 times. There could be another lens somewhere here that could be saying 10 times. Okay. So usually there's about 3 objective lenses in a typical microscope. It could be 1, it could be 2, or it could be 3, right? So you can write an equation if you take a normal microscope about the total magnification. Okay, about the total magnification of the entire microscope. You can say total magnification, yes, is equal to what? Magnification of the objective lens magnification of objective lens multiplied by magnification of eyepiece lens. Okay. So now usually your eyepiece lens has a fixed magnification, usually around 10 times. So the objective lenses usually have 10 times, 40 times and 100 times. Okay. So usually you can write here as a star mark, can write usually maximum magnification of a compound light microscope is around 1000 times, because why? 10 times into 100 is around 1000 times. Okay, is around 1000 times for a typical compound light microscope. Okay, can you write this? Total magnification is equal to magnification of the objective lens multiplied by magnifications of the eyepiece lens. Maximum magnification of a compound light microscope is around 1000 times. Okay, 1000 times. Very good eyesight. Okay. Any questions? You can ask anything. Okay, right. What do you want me to zoom out again now? Okay. Okay, so compound light microscope is kind of like this. So in the practical seminar video, I describe all the parts with a real microscope, which is, don't ask me how I got it. It's not a very old microscope that I have. Okay, so you kind of demonstrate all the parts using the real microscope, so you can take a look. Okay, definitely you have to watch, huh? Otherwise you guys will be telling I didn't explain the parts of the microscope. Okay, you have to watch the recording. Okay, you have to promise that, otherwise this won't be successful flash up to speed program. Okay, I'll be sending it tonight itself. With you have a week to watch it. Okay, baranda morning. Okay, how long is the video? Because it's practicals 3 hours 21 minutes. With all the organic molecule identification, and also with the time including I have given with the time I give to write. So you can fast forward those parts where I give time to write down, draw diagrams. You can fast forward. It is then around 2.5 hours, maybe 2 hours if you remove all the time I give to draw diagrams and other things. It's then about 2 hours. Okay, right? Because I heat the things and wait for the result to come out, and then I then talk around. I give an interval. There's the interval in the video. Yes, the interval is also in the video. So the interval is around 20 minutes. No? So interval is also in the video. So yeah, maximum 2 hour video. Okay. Because I couldn't be bothered to crop out the interval. Okay, you can take an interval at that time or something. Okay, then we talked about how resolution power is limited by the wavelength. Then you need to know what is the resolution power of the maximum resolution power. That means after you reach this minimum size of the specimen, there is no use trying to observe it by the light microscope. Okay, the resolution power, or the maximum resolution power of the light microscope, okay, is about 0.2 micrometers. So where is 0.2 micrometers? You can see it is kind of within 1 micrometer is the mitochondria. Then sort of below that you have around here, before 100 nanometers, you have somewhere here, no, 0.2 micrometers is the resolution power of the compound light microscope. You can see exactly that is where you stop using light microscope. That is because beyond that point you can magnify. Beyond that point you can magnify, but the image is not clear. The image is not clear because why? The wavelength of light is too long. The image is going to be blurry. Got it? So you can write here. I don't have much space, so please write with here max resolution power of compound light microscope is how much? 0.2 micrometers. The maximum resolution power of the compound light microscope is 0.2 micrometers. Write down the maximum resolution power is 0.2 micrometers. Okay, so as with any problem with the, as with any problem, when you when you encounter a certain problem, you need to find a solution to that, right? So you can't give up your study on cytology just because the resolution power of the light microscope is this much, right? You have to do something, no? You have to observe cells, you have to improve cytology. Got it? So scientists thought about it and they decided to use a different type of wave altogether. Okay, that is this, a different type of wave they decided to use. You can see here, they decided to use electrons. They decided to use electrons. So electrons, if you are using as a wave to observe a specimen, what can you do is, you have done cathode ray experiment. Have you done? For chemistry, you did, no? Atomic structure is all over, right? You have done cathode ray experiment, so electrons can have particulate as well as wave-like properties, no? Yeah. So what you can do is, if you are using electrons as a waveform to observe specimen, using, electrons are negatively charged and also they have a magnetic charge as well, no? I think you have put a magnet and the electron wave bends perpendicularly. Home story right? Cathode ray experiment again. Into the cathode ray tube, you put a magnet and it bends perpendicularly or something, because electrons are magnetic, right? Electrons are also magnetic. So imagine you are going to use electrons as your wave, then how can you manipulate them? How can you manipulate them? How can you change them? You can use a magnet. You can use a magnet, a series of magnets actually, to change the wavelengths of these electrons. So that's why you can see, if you check this, you don't have actually lenses. You can cut it here in this diagram. You can cut it. These are not lenses. These are, right, magnetic magnets. So you can say magnetic lenses. In the light microscope, they were glass lenses. These were magnets. Okay, magnetic lenses are used to focus the light. Okay, magnetic lenses are used to focus the light. Therefore, electron source is there. There are several magnets to condense. And where is the specimen? Specimen. This is the specimen. They go through the specimen, and later you can observe using a computer screen, or you can directly observe by, not directly observe. The electrons are going to fold on a digital detector or a photographic film, and you can take a look at the photographic film. But in modern electron microscopes, there's a computer screen. So there's a software that can take a look at the electrons and will generate a computer image screen, so that you can see it with your, you can never see it directly, but you can observe it through a photographic film of electrons, or you can see it on a computer screen. Okay. Okay, come to the resource book. Let's read about it. The electron microscope. Okay, let's read about it. The electron microscope. Limitation imposed upon the resolution power of the light microscope is by the wavelength of light. Can you highlight? Limitation is imposed upon the resolution power is by the wavelength of light. Highlight that. Then resolution power is inversely proportional to wavelength. Highlight that also. Resolution power is inversely proportional to wavelength. That's what I gave you here, right? When the resolution power increases, wavelength decreases. Due to this, scientists consider the use of other forms of radiation with comparatively shorter wavelengths. Okay. As a result, electron microscopes were developed. In electron microscopy, a beam of electrons are focused through the specimen or onto its surface. Okay. In practice, electron microscope magnifies about 5 into 10 to the power 5 times. That means 500,000 times. Ne? Lakshapa, 5 lakhs. Highlight. Magnification is about 5 into 10 to the power 5. Resolution about 2 nanometers. So here, What was the resolution? 0.2 micrometers. But with a electron microscope, you can go to about 2 nanometers. Can you see, Nidhi? The limit of electron microscopy is about 2 nanometers. Right? Okay, right. Then electron microscopes have revealed many organisms and other subcellular structures that were impossible to resolve with light microscopes. Okay, so now we are going to do this comparison between light versus electron microscope. Okay, write down on this side light microscope. On this side, write down electron microscope. We are going to do a comparison. Okay. So what we are going to do is actually I want you to write a certain criteria in front. So for example, focusing in light microscope is done by what? Glass lenses. Focusing is by glass lenses. In electron microscope, you use powerful magnets to focus the beam of electrons. Okay, powerful magnets. Powerful magnets are used. Then detection, that means how you can see the image. Okay, detected by naked eye. Naked eye means you don't need to use any instrument, you can just put your eye and see by naked eye. Okay, but can you see electrons with your naked eye? Your eyes are photoreceptors, you can only see light with your eyes. Okay, so you need some instrument to see the electrons. Okay, so we earlier we used micrographs, they are like our photographic plates, right? Micrographs, but now we use computer screens, but you can say micrographs. Then the thing is, when you use the electron microscope, because electrons are, they are negatively charged, and they are, when these electrons go through a specimen, imagine you have a piece of tissue, for example, by during a surgery you found out a cancer, and you take a slice of that cancer and put it under the electron microscope to see, right? When electrons are going through that, the specimen is going to be killed. The specimen cells are going to be killed because of the electrons. But in the light microscope, because you are observing just by visible light, no? So the specimen cells won't be killed. Okay, that's why you can observe both. I mean, you can observe some dead tissue, coke cells, right? You can observe they are dead cells, no? Right? Living and non-living both can observe under the light microscope. Living and non-living. So please understand, okay, you don't have to use only non-living things. But if you take a non-living specimen, if you put it under the electron microscope, it is going to be killed by the electron microscope. Got it? It is going to be killed by the electron microscope. That's why we say only non-living. Okay, write with me. Okay, then what do you mean by color? Color means color is due to different wavelengths of visible light, no? So in electron microscopy there is no visible light, no? Actual color observed But in electron microscopy, since it's electrons, you can't see actual color. Cannot. Then you will understand that even though, I mean, for example, if you put some bacteria under the light microscope and you try to observe, the bacteria actually very transparent. You don't have much color, because why? They are very small organisms. So almost all the light is just going to go through them. So they just appear with some only with an outline, right? They'll appear almost transparent. If you want to see them very clearly, some of the structures, we have to put some artificial dyes. Okay, a gram stain, or there are some dyes. So you can use some dyes to see some color. But there's no use in putting those dyes. Dyes are what? They are color pigments. There is no use in putting those onto a electron microscope specimen. But you can use heavy metals, because through some heavy metals, uh, the electrons go in different ways, so that you can observe a bit of difference. I mean, difference in the sort of the, it's all, I mean, it's obviously it's black and white. You can see some dark areas, light areas, when you use heavy metals. Okay, quickly do this comparison. Taggal liya again. Okay. Now for the, so we'll finish for today with a comparison between different types of electron microscopes. Okay, right? So I have given the two types of electron microscopes that you have in your syllabus, that is scanning as well as transmission electron microscopes. Let's go through the resource. Okay, now people listen, this is how we are going to go through. You already know how I am doing the normal theory class. We rely on the resource book as our main sort of information source. Then I explain it. Okay, then Together we highlight, then we highlight definitions, important points, then we write down a short note, then we do questions, then I give you homework essays. This is how it's going to go. Okay. This is the most time effective as well as the most interactive way of doing biology that I have discovered over, I mean, as a student, as a teacher, in both ways. Okay. There is no use giving extensive notes because you are not going to need 2 notes to study. Then there is no use in just reading the resource without drawing or doing questions or making a short note, because that is again going to be not productive. Okay. So whenever you are reading the resource with me, I want you to pay 100% attention and highlight everything that you need. Okay. Maintain the resource book nicely. Okay, so page number 23, transmission electron microscope. Let's read it. It is used to study internal structures of cells. In this microscope, a beam of electrons is passed through a thin, especially prepared slice of material. Okay, highlight. A beam of electrons pass through. A beam of electrons is passed through. Right? So that phrase is very important. Then highlight, a very thin specimen is used. You need a very thin specimen. Specimens stained with heavy metals which attach more to certain cellular structures than other areas. So use heavy metals before to prepare the specimen. Image reflects the pattern of electrons passing through the specimen displayed on a screen. While electrons pass through the specimen, more electrons get displayed on regions where densely stained. So you know, heavy metals usually adhere to some structures more. Could be mitochondria, could be certain internal membranes. Got it? So when the electrons pass through, they go through the heavy metals more, because why? Electrons like to go through metallic substances. Okay, so that when you put it into a screen, you can see it. You can see the places where many electrons went through, so that will appear in dark color. Okay, right. So that is transmission. On this side, I have given transmission electron microscope, EM. Okay, let's read about the scanning one as well. Then we are going to do a comparison. Scanning electron microscope. Okay. In this instrument, a fine beam of electrons. Is reflected from the surface. Can you highlight reflected from the surface of the specimen? Specimen is coated with gold. Highlight that also, coated with gold. Here the specimen scatters many electrons, whereas others are absorbed. Okay. This is ideal to observe the surface view in 3D appearance. Highlight, ideal to observe surface view in 3D. Okay, let's see. On the scanning side, I can write electrons are reflected. On the transmission side, I can say electrons pass through. Electrons pass through a very thin specimen. Electrons pass through a very thin specimen. Next, on the scanning side, you can write coated with gold. You can say on this side stained with heavy metals. Kattiya not sleeping neither. Got it? I know 4 hours is a long time. Then scanning side, you can say used to observe surface view in 3D. On the transmission side, you can write to study internal structure of cells. To study internal structure of cells. Okay, then finally on the scanning side you can write this is something additional. Okay, specimen scatters many electrons. That is because through the gold many electrons don't go through. Gold specimen scatters many electrons and others are absorbed. Specimen scatters many electrons and others are absorbed. Then on the transmission side, final point, more electrons displayed in regions where structures are densely stained. चले. Okay, read with me. On the scanning side, electrons are reflected from the surface. Transmission side, electrons pass through a very thin specimen. On the scanning side, the specimen is coated with gold. The transmission side, stained with heavy metals. On the scanning side, used to observe surface view in 3D. On the transmission side, used to observe or used to study internal structure of cells. On the scanning side, specimen scatters many electrons and others are absorbed. On the transmission side, more electrons are displayed in regions where structures are densely stained by the heavy metal. No? Okay. Right. So we'll stop there for the day. Okay. I was actually planning to finish plasma membrane as well. Okay. So from next week, please start joining by 5:50. Okay. So then I can start at exactly around 6. Okay. So I hope the people who are remaining, that's good. But the others will watch the recording. I hope. Then that is going to be the end of up to speed sessions day 3. Okay, so hope you are doing well in the theory class. If you have any issue, you can contact about the theories, right? Any doubts about the theory parts that you have, you can contact me either through the hotline, or you can ask me physically during the class, or send a Zoom to the Zoom chat. I will explain. Okay, so yeah, we'll stop for the day. Then any issues? Thank you for joining then, and Keep studying a bit. Okay. Yep. Have a good night. Okay, you're welcome. Okay, good night. Good night. Thank you. Okay. Welcome. Good night. Keep studying a bit. Okay. Okay. Good night.